Hello, and welcome to the Quiet Science Channel. I'm so glad you found your way here tonight. Maybe you've had a long day. Maybe you're the kind of person who likes drifting off to deep thoughts about the universe rather than counting sheep. Either way, you're in the right place. Tonight, we're exploring one of the most profound questions humans have ever asked. Not where did we come from in the sense of our immediate ancestors, our grandparents, or great-grandparents. We're going much further back than that. We're going back 4 billion years to a time when Earth was barely recognizable, when the planet was more hell than home, when the very air would have poisoned us in seconds. We're asking the question that has puzzled philosophers, theologians, and scientists for thousands of years. How did life begin? Before we drift into that ancient story together, if these calm, curious explorations help you unwind at the end of your day or give you something peaceful to think about as you fall asleep, consider liking this video and subscribing to the channel. It helps others find their way here too, one restful soul at a time. Now, let your breathing slow. Let your mind settle. We're about to travel back to a world that no longer exists, to witness, as best we can, the moment when lifeless chemistry became living biology. The moment when atoms and molecules, scattered across a cooling planet, somehow organized themselves into something that could grow, reproduce, and eventually ask questions about its own origins. This is the story of Earth's first spark, the story of how life began. Think about what life actually is for a moment. Right now, as you listen to these words, trillions of cells in your body are carrying out chemical reactions. Your heart beats without you thinking about it. Your lungs expand and contract. Neurons fire across your brain, creating thoughts, memories, awareness. All of this happens because, billions of years ago, something extraordinary occurred on a young, violent planet. The origin of life, what scientists call abiogenesis, is the study of how life arose from non-living matter. It's the investigation into how simple chemicals, just carbon and hydrogen and nitrogen and oxygen, arrange themselves in such specific ways that they could replicate, could respond to their environment, could evolve. It's the question of how we got from a sterile rock to a planet teeming with bacteria, algae, plants, animals, and eventually, conscious beings capable of pondering their own existence. For most of human history, this question was answered with mythology, with creation stories that varied from culture to culture, but shared a common thread. Life, these stories said, came from the gods, from divine intervention, from forces beyond the natural world. And while these stories provided comfort and meaning, they didn't provide testable explanations. They didn't allow us to peer backward through time and understand the actual mechanisms, the actual chemistry, the actual physics that transformed a dead planet into a living one. That began to change in the 1800s. Charles Darwin, contemplating evolution and the diversity of life, wrote to a friend about what he called a warm little pond, a place where the right chemicals in the right conditions might have combined to form the first protocell, the first living thing. He knew better than to publish these speculations. He knew that the origin of life was an even more contentious topic than evolution itself. But the seed was planted. If life could evolve, if species could change over time, then perhaps life itself had a beginning that could be understood through natural processes. In the 1920s, two scientists working independently arrived at similar conclusions. Alexander Oparin in Russia and John Haldane in Britain both proposed that Earth's early atmosphere was very different from today's. They suggested that it was a reducing atmosphere, filled with gases like methane, ammonia, and hydrogen, and that in such an environment, with the right energy sources like lightning or ultraviolet light from the sun, simple organic molecules could form. These molecules, they theorized, would accumulate in Earth's oceans, creating what Haldane memorably called a primordial soup, a rich broth of organic compounds from which life could emerge. This was the Oparin-Haldane hypothesis, and it transformed the study of life's origins from philosophical speculation into a scientific question. If their ideas were correct, if simple molecules could indeed form from inorganic materials under the right conditions, then perhaps the process could be replicated in a laboratory. Perhaps we could recreate, in miniature, the conditions that gave rise to life on Earth. That's exactly what happened in 1952, when a young graduate student named Stanley Miller built an apparatus of glass flasks and tubes, filled it with water and gases meant to mimic the early Earth, and sent electrical sparks crackling through the mixture. Within days, the water in his experiment had turned brown. When analyzed, it contained amino acids, the building blocks of proteins, the very molecules that make up the machinery of every living cell. Miller's experiment made headlines around the world. It seemed to prove that life's building blocks could form spontaneously from simple chemicals. It suggested that the gap between non-life and life might not be as vast as previously thought. It opened a field of research that continues today, with scientists around the world working to understand how chemistry became biology, how molecules became life. But as we'll discover tonight, the story is far more complex and far more beautiful than those early experiments suggested. We've learned that Earth's early atmosphere was probably different than Miller assumed. We've discovered hydrothermal vents at the bottom of the ocean, underwater volcanoes that spew hot, mineral-rich water, and that might have provided the perfect environment for life's emergence. We've found evidence that RNA, the molecule closely related to DNA, might have come before proteins, acting as both genetic material and catalyst in an ancient RNA world. We've traced the genetic code backward through evolution to identify LUSA, the last universal common ancestor, a microorganism that lived around 4.2 billion years ago and was the ancestor of every living thing on Earth today, from bacteria to blue whales, from oak trees to humans. We've found fossils, stromatolites, layered rocks created by microbial mats that preserve evidence of life from 3.5 billion years ago. And yet, despite all this progress, despite decades of research and countless experiments, we still don't know exactly how life began. We have pieces of the puzzle. We understand many of the chemical reactions that must have occurred. We can point to plausible environments where these reactions might have taken place, but the complete picture, the step-by-step pathway from chemicals to cells, remains elusive. The origin of life is still one of science's greatest mysteries. That's what makes it so fascinating. That's what makes it perfect for a quiet night of contemplation. We're not just learning about ancient history. We're exploring one of the fundamental questions about existence itself. We're asking what it means to be alive, what separates the living from the non-living, and whether life is a cosmic accident or an inevitable consequence of chemistry and physics. So, settle in. Let your mind wander back through deep time. We're about to visit an Earth that was nothing like the one we know today. An Earth of molten rock and toxic gases, of meteorite impacts and volcanic eruptions, of a sun that was dimmer but a sky that was filled with more violence. It was in this crucible, this hellish landscape, that life somehow found a way to begin. Word count for section 1. 1,380 words. Let me continue expanding to reach 1,600 words. The question of life's origin is intimately connected with the story of Earth itself. Our planet formed 4.6 billion years ago from a disk of dust and gas orbiting the young sun. Through a process called accretion, tiny particles stuck together, forming larger and larger clumps, eventually creating rocky planetesimals that collided and merged. One of these collisions was particularly dramatic. A Mars-sized object that scientists call Theia slammed into the proto-Earth in a cataclysmic impact that melted both bodies and splashed debris into orbit. That debris eventually coalesced to form our moon. In those first few 100 million years, Earth was a world of fire. The surface was molten. Volcanoes erupted constantly, spewing gases that would become the atmosphere. Meteorites and asteroids bombarded the surface in what scientists call the late heavy bombardment, impacts so frequent and so violent that they would have vaporized any oceans and sterilized the surface. It seemed impossible that life could emerge in such conditions. And yet, as we'll see, recent evidence suggests that life may have begun even earlier than we thought, perhaps as early as 4.2 billion years ago, only a few 100 million years after Earth formed. This raises a profound question. If life emerged so quickly on Earth, almost as soon as conditions permitted, what does that tell us about life in the universe? Is life common, arising wherever liquid water and the right chemicals come together? Or was Earth's early life a cosmic fluke, a 1 in a trillion chance that happened to occur on our particular planet? These questions drive the field of astrobiology, the search for life beyond Earth. Because if we can understand how life began here, we'll be better equipped to look for it elsewhere, on Mars, on the moons of Jupiter and Saturn, on the thousands of exoplanets orbiting distant stars. But first, we need to understand what happened here, on our own world, in Earth's distant past. We need to piece together the story from fragments, from ancient rocks and chemical experiments, from computer simulations and genetic analyses. It's detective work on a cosmic scale, and every new discovery brings us closer to answering that oldest of questions. How did life begin? To understand how life began, we first need to understand the world in which it emerged. And that world, 4 billion years ago, was nothing like the Earth we know today. Scientists call this period the Hadean Eon, named after Hades, the Greek god of the underworld. The name is fitting. If you could somehow travel back to Earth 4.5 billion years ago, you would find yourself in a landscape that resembles our worst visions of hell, far more than it resembles the blue-green planet we call home. The Hadean Eon spans from Earth's formation, about 4.6 billion years ago, to roughly 4 billion years ago. That's 600 million years, longer than the entire time from the Cambrian explosion to today, longer than all of complex life on Earth. And throughout most of that time, our planet was, by any reasonable definition, uninhabitable. Imagine standing on the surface of Hadean Earth. Not that you could, of course. There's no solid ground to stand on, only a roiling ocean of molten rock. The surface temperature exceeds 2,000 degrees Celsius. The sky glows red and orange from the heat radiating from below. Above you, if you could somehow survive long enough to look up, the sky would be dark despite the sun being up, choked with rock vapor and carbon dioxide. The air itself is poison, a thick atmosphere of gases that would kill any modern organism instantly. There's no oxygen. The atmosphere is instead filled with water vapor, carbon dioxide, nitrogen, methane, and ammonia. And every so often, sometimes as frequently as a few times per century, the sky lights up as another asteroid or comet slams into the planet. Some of these impacts are small, merely adding to the constant bombardment that keeps the surface molten. But some are massive, planet-altering events. The largest impacts vaporize entire oceans, if oceans have managed to form at all, and send plumes of vaporized rock into space. The impacts keep the surface temperature high, prevent any stable crust from forming, ensure that Earth remains a ball of molten chaos. This is the late, heavy bombardment, a period around 4 billion years ago when the inner solar system was swept clean of debris by the gravitational influence of the giant planets. Jupiter and Saturn, still migrating in their orbits, flung countless asteroids and comets toward the inner planets. Earth, being larger and having more gravity than Mercury, Venus, or Mars, caught more than its fair share. The surface was repeatedly sterilized by impacts. If life had somehow managed to begin during this time, it would have been destroyed over and over, reset to zero by each major collision. Or so scientists thought for many years. But recent evidence has challenged this picture. It turns out that Earth may have been more hospitable, more quickly, than we previously believed. The key evidence comes from tiny crystals of a mineral called zircon. Zircons are remarkable little time capsules. They're extremely hard, resistant to erosion and chemical alteration, and they incorporate uranium into their crystal structure. The uranium decays into lead at a known rate, which allows scientists to determine precisely how old each zircon crystal is. Some of the oldest materials on Earth are zircons found in the Jack Hills of Western Australia. These crystals are over 4 billion years old, formed during the Hadean Eon, and they've survived everything Earth could throw at them across 4 billion years of geological time. But what's remarkable about these ancient zircons isn't just their age. It's what they tell us about the conditions under which they formed. Zircons form in molten rock, in magma deep underground, or in volcanic eruptions. But the chemical composition of these ancient zircons suggests something unexpected. They seem to have formed in the presence of liquid water. The ratio of isotopes within the crystals indicates that the magma from which they crystallized had interacted with water. This implies that Earth had liquid water, and therefore oceans, as early as 4.4 billion years ago, only about 200 million years after the planet formed. This discovery revolutionized our understanding of the Hadean. Instead of an entirely molten hellscape, we now think that Earth cooled relatively quickly. Within a few hundred million years, the surface had solidified enough to form a crust. Water vapor in the atmosphere condensed and fell as rain, torrential downpours that may have lasted for thousands of years, gradually filling the first oceans. The planet was still violent, still experiencing frequent impacts and volcanic eruptions, but it wasn't entirely uninhabitable. There were places, perhaps many places, where liquid water existed in stable pools and seas. This matters enormously for the story of life's origin. Life, as we know it, requires liquid water. Every biochemical reaction that sustains life takes place in water. Proteins fold in water. DNA replicates in water. Cells are bags of water with membranes around them. If Earth had liquid water 4.4 billion years ago, then the conditions for life could have existed almost from the very beginning, and there's evidence that they did. In rocks from Western Australia, scientists have found carbon isotopes in minerals that formed 4.1 billion years ago. These carbon isotopes show the characteristic signature of biological processes, the preference for lighter isotopes that living organisms display. This doesn't prove that life existed that early. The carbon signatures could potentially have formed through non-biological processes, but it's suggestive. It's consistent with the idea that life might have emerged very early in Earth's history. The Hadean atmosphere was nothing like the one we breathe today. There was virtually no oxygen. The sky would have looked different, perhaps more hazy, perhaps tinged with colors we don't associate with Earth's sky. The sun was about 30% dimmer than it is today, but Earth wasn't frozen because of the thick greenhouse atmosphere. Carbon dioxide and methane trapped heat, keeping the surface temperature above the freezing point of water despite the dimmer sun. This early atmosphere probably came from two sources. Some of it was primordial, captured from the solar nebula when Earth formed, mostly hydrogen and helium. But most of that light gas quickly escaped to space. The more important source was volcanic outgassing. As the planet's interior sorted itself out, with heavy iron sinking to form the core and lighter rocks rising to form the mantle and crust, volcanoes released enormous quantities of gas from the interior. These gases, water vapor, carbon dioxide, nitrogen, and smaller amounts of methane, ammonia, hydrogen sulfide, and others, formed the secondary atmosphere that would persist for billions of years. The Hadean was also when Earth's magnetic field strengthened. Deep in the planet's core, liquid iron swirls and flows, creating electrical currents that generate a magnetic field. This field extends far beyond Earth's surface, creating the magnetosphere, an invisible shield that deflects charged particles from the sun. Without this magnetic field, the solar wind would gradually strip away Earth's atmosphere, as has happened on Mars. The magnetic field was crucial for maintaining an atmosphere capable of supporting life. By the end of the Hadean, around 4 billion years ago, Earth had transformed. The bombardment had slowed. The crust had stabilized. Continents, small ones at first, had begun to form through volcanic activity and the collision of crustal plates. The oceans were extensive, covering most of the planet's surface. The atmosphere was thick and reducing, rich in gases that could react to form organic molecules. It was still not a hospitable place by our standards. The air was unbreathable. The water was likely hot, perhaps close to boiling in many places. The sky was dominated by volcanic eruptions and lightning storms. Ultraviolet radiation from the sun, unfiltered by an ozone layer that wouldn't exist for billions of years, bombarded the surface. But it was a world where chemistry could happen, where molecules could form and react and recombine, where energy was abundant from the sun, from lightning, from volcanic heat, from the energy released by chemical reactions themselves. It was a world on the edge, balanced between chaos and order, between sterility and the possibility of something more. It was in this world, during the late Hadean or the early Archean that followed, that life somehow began. The exact when and where remain uncertain. But the stage was set. The ingredients were present. All that was needed was the right combination, the right sequence of events, the right bit of chemistry in the right place at the right time. And somewhere on that ancient Earth, in some warm pool or deep sea vent or mineral-rich tidal flat, it happened. Lifeless molecules crossed that mysterious boundary and became something that could be called alive. The geological processes of the Hadean were fundamentally different from those we observe today. Without continents and with a thinner crust, plate tectonics may have operated differently if it operated at all. Some scientists suggest that the Hadean crust was more like a stagnant lid, with volcanism occurring through hot spots rather than at plate boundaries. Others propose that a primitive form of plate tectonics began very early, as soon as the crust cooled enough to crack and move. Evidence for these early geological processes is frustratingly scarce. The problem is that the rock cycle destroys its own history. Rocks formed in the Hadean have been melted, metamorphosed, eroded, and recycled countless times over 4 billion years. Only the hardiest minerals, like those zircon crystals, survive to tell the tale. Most of the Hadean is a blank page in Earth's geological record. But we can make inferences from what we do know. We know that Earth's interior was hotter in the Hadean. The planet had recently formed through the collision of smaller bodies, and all that kinetic energy had been converted to heat. Additionally, there were far more radioactive elements in Earth's interior, elements like uranium and thorium that decay over time, releasing heat. This meant more volcanic activity, more magma rising from the mantle, more heat flow from the interior to the surface. This heat had consequences for any emerging life. On one hand, high temperatures are generally inhospitable to life. Most organisms can't survive in boiling water, let alone the much hotter temperatures found near volcanic vents. But on the other hand, heat provides energy, and energy is necessary for the chemical reactions that underlie life. Some of Earth's most ancient organisms, based on genetic analyses, appear to have been thermophils, heat-loving organisms that thrived in hot water. This suggests that high temperatures might not have been a barrier to early life, but rather a requirement. The oceans of the Hadean were probably different from modern oceans in another important way. They were likely more acidic. Carbon dioxide dissolves in water to form carbonic acid, and with the thick carbon dioxide atmosphere, the early oceans probably had a pH closer to that of vinegar than to modern seawater. This acidity would have implications for what kinds of chemistry could occur, what minerals would dissolve or precipitate, what kinds of molecular structures would be stable. Hello, and welcome to the Quiet Science channel. I'm so glad you found your way here tonight. Maybe you've had a long day. Maybe you're the kind of person who likes drifting off to deep thoughts about the universe rather than counting sheep. Either way, you're in the right place. Tonight, we're exploring one of the most profound questions humans have ever asked. Not where did we come from, in the sense of our immediate ancestors, our grandparents or great-grandparents. We're going much further back than that. We're going back 4 billion years, to a time when Earth was barely recognizable, when the planet was more hell than home, when the very air would have poisoned us in seconds. We're asking the question that has puzzled philosophers, theologians, and scientists for thousands of years. How did life begin? Before we drift into that ancient story together, if these calm, curious explorations help you unwind at the end of your day or give you something peaceful to think about as you fall asleep, consider liking this video and subscribing to the channel. It helps others find their way here too, one restful soul at a time. Now, let your breathing slow. Let your mind settle. We're about to travel back to a world that no longer exists, to witness, as best we can, the moment when lifeless chemistry became living biology. The moment when atoms and molecules, scattered across a cooling planet, somehow organized themselves into something that could grow, reproduce, and eventually ask questions about its own origins. This is the story of Earth's first spark, the story of how life began. Think about what life actually is for a moment. Right now, as you listen to these words, trillions of cells in your body are carrying out chemical reactions. Your heart beats without you thinking about it. Your lungs expand and contract. Neurons fire across your brain, creating thoughts, memories, awareness. All of this happens because billions of years ago, something extraordinary occurred on a young, violent planet. The origin of life, what scientists call abiogenesis, is the study of how life arose from non-living matter. It's the investigation into how simple chemicals, just carbon and hydrogen and nitrogen and oxygen, arranged themselves in such specific ways that they could replicate, could respond to their environment, could evolve. It's the question of how we got from a sterile rock to a planet teeming with bacteria, algae, plants, animals, and eventually, conscious beings capable of pondering their own existence. For most of human history, this question was answered with mythology, with creation stories that varied from culture to culture, but shared a common thread. Life, these stories said, came from the gods, from divine intervention, from forces beyond the natural world. And while these stories provided comfort and meaning, they didn't provide testable explanations. They didn't allow us to peer backward through time and understand the actual mechanisms, the actual chemistry, the actual physics that transformed a dead planet into a living one. That began to change in the 1800s. Charles Darwin, contemplating evolution and the diversity of life, wrote to a friend about what he called a warm little pond, a place where the right chemicals, in the right conditions, might have combined to form the first protocell, the first living thing. He knew better than to publish these speculations. He knew that the origin of life was an even more contentious topic than evolution itself, but the seed was planted. If life could evolve, if species could change over time, then perhaps life itself had a beginning that could be understood through natural processes. In the 1920s, two scientists working independently arrived at similar conclusions. Alexander Oparin in Russia and John Haldane in Britain both proposed that Earth's early atmosphere was very different from today's. They suggested that it was a reducing atmosphere, filled with gases like methane, ammonia, and hydrogen, and that in such an environment, with the right energy sources like lightning or ultraviolet light from the sun, simple organic molecules could form. These molecules, they theorized, would accumulate in Earth's oceans, creating what Haldane memorably called a primordial soup, a rich broth of organic compounds from which life could emerge. This was the Oparin-Haldane hypothesis, and it transformed the study of life's origins from philosophical speculation into a scientific question. If their ideas were correct, if simple molecules could indeed form from inorganic materials under the right conditions, then perhaps the process could be replicated in a laboratory. Perhaps we could recreate, in miniature, the conditions that gave rise to life on Earth. That's exactly what happened in 1952, when a young graduate student named Stanley Miller built an apparatus of glass flasks and tubes, filled it with water and gases meant to mimic the early Earth, and sent electrical sparks crackling through the mixture. Within days, the water in his experiment had turned brown. When analyzed, it contained amino acids, the building blocks of proteins, the very molecules that make up the machinery of every living cell. Miller's experiment made headlines around the world. It seemed to prove that life's building blocks could form spontaneously from simple chemicals. It suggested that the gap between non-life and life might not be as vast as previously thought. It opened a field of research that continues today, with scientists around the world working to understand how chemistry became biology, how molecules became life. But as we'll discover tonight, the story is far more complex and far more beautiful than those early experiments suggested. We've learned that Earth's early atmosphere was probably different than Miller assumed. We've discovered hydrothermal vents at the bottom of the ocean, underwater volcanoes that spew hot, mineral-rich water, and that might have provided the perfect environment for life's emergence. We found evidence that RNA, the molecule closely related to DNA, might have come before proteins, acting as both genetic material and catalyst in an ancient RNA world. We've traced the genetic code backward through evolution to identify Luca, the last universal common ancestor, a microorganism that lived around 4.2 billion years ago and was the ancestor of every living thing on Earth today, from bacteria to blue whales, from oak trees to humans. We've found fossils, stromatolites, layered rocks created by microbial mats that preserve evidence of life from 3.5 billion years ago. And yet, despite all this progress, despite decades of research and countless experiments, we still don't know exactly how life began. We have pieces of the puzzle. We understand many of the chemical reactions that must have occurred. We can point to plausible environments where these reactions might have taken place. But the complete picture, the step-by-step pathway from chemicals to cells, remains elusive. The origin of life is still one of science's greatest mysteries. That's what makes it so fascinating. That's what makes it perfect for a quiet night of contemplation. We're not just learning about ancient history, we're exploring one of the fundamental questions about existence itself. We're asking what it means to be alive, what separates the living from the non-living, and whether life is a cosmic accident or an inevitable consequence of chemistry and physics. So settle in. Let your mind wander back through deep time. We're about to visit an Earth that was nothing like the one we know today. An Earth of molten rock and toxic gases, of meteorite impacts and volcanic eruptions, of a sun that was dimmer, but a sky that was filled with more violence. It was in this crucible, this hellish landscape, that life somehow found a way to begin. The question of life's origin is intimately connected with the story of Earth itself. Our planet formed 4.6 billion years ago from a disk of dust and gas orbiting the young sun. Through a process called accretion, tiny particles stuck together, forming larger and larger clumps, eventually creating rocky planetesimals that collided and merged. One of these collisions was particularly dramatic. A Mars-sized object that scientists call Theia slammed into the proto-Earth in a cataclysmic impact that melted both bodies and splashed debris into orbit. That debris eventually coalesced to form our moon. In those first few 100 million years, Earth was a world of fire. The surface was molten. Volcanoes erupted constantly, spewing gases that would become the atmosphere. Meteorites and asteroids bombarded the surface in what scientists call the late heavy bombardment, impacts so frequent and so violent that they would have vaporized any oceans and sterilized the surface. It seemed impossible that life could emerge in such conditions. And yet, as we'll see, recent evidence suggests that life may have begun even earlier than we thought, perhaps as early as 4.2 billion years ago, only a few 100 million years after Earth formed. This raises a profound question. If life emerged so quickly on Earth, almost as soon as conditions permitted, what does that tell us about life in the universe? Is life common, arising wherever liquid water and the right chemicals come together? Or was Earth's early life a cosmic fluke, a 1 in a trillion chance that happened to occur on our particular planet? These questions drive the field of astrobiology, the search for life beyond Earth. Because if we can understand how life began here, we'll be better equipped to look for it elsewhere, on Mars, on the moons of Jupiter and Saturn, on the thousands of exoplanets orbiting distant stars. But first, we need to understand what happened here, on our own world, in Earth's distant past. We need to piece together the story from fragments, from ancient rocks and chemical experiments, from computer simulations and genetic analyses. It's detective work on a cosmic scale, and every new discovery brings us closer to answering that oldest of questions. How did life begin? To understand how life began, we first need to understand the world in which it emerged. And that world, 4 billion years ago, was nothing like the Earth we know today. Scientists call this period the Hadean Eon, named after Hades, the Greek god of the underworld. The name is fitting. If you could somehow travel back to Earth 4.5 billion years ago, you would find yourself in a landscape that resembles our worst visions of hell far more than it resembles the blue-green planet we call home. The Hadean Eon spans from Earth's formation, about 4.6 billion years ago, to roughly 4 billion years ago. That's 600 million years, longer than the entire time from the Cambrian explosion to today, longer than all of complex life on Earth. And throughout most of that time, our planet was, by any reasonable definition, uninhabitable. Imagine standing on the surface of Hadean Earth. Not that you could, of course. There's no solid ground to stand on, only a roiling ocean of molten rock. The surface temperature exceeds 2,000 degrees Celsius. The sky glows red and orange from the heat radiating from below. Above you, if you could somehow survive long enough to look up, the sky would be dark despite the sun being up, choked with rock vapor and carbon dioxide. The air itself is poison, a thick atmosphere of gases that would kill any modern organism instantly. There's no oxygen. The atmosphere is instead filled with water vapor, carbon dioxide, nitrogen, methane, and ammonia. And every so often, sometimes as frequently as a few times per century, the sky lights up as another asteroid or comet slams into the planet. Some of these impacts are small, merely adding to the constant bombardment that keeps the surface molten. But some are massive, planet-altering events. The largest impacts vaporize entire oceans, if oceans have managed to form at all, and send plumes of vaporized rock into space. The impacts keep the surface temperature high, prevent any stable crust from forming, ensure that Earth remains a ball of molten chaos. This is the late heavy bombardment, a period around 4 billion years ago, when the inner solar system was swept clean of debris by the gravitational influence of the giant planets. Jupiter and Saturn, still migrating in their orbits, flung countless asteroids and comets toward the inner planets. Earth, being larger and having more gravity than Mercury, Venus, or Mars, caught more than its fair share. The surface was repeatedly sterilized by impacts. If life had somehow managed to begin during this time, it would have been destroyed over and over, reset to zero by each major collision. Or so scientists thought for many years. But recent evidence has challenged this picture. It turns out that Earth may have been more hospitable, more quickly, than we previously believed. The key evidence comes from tiny crystals of a mineral called zircon. Zircons are remarkable little time capsules. They're extremely hard, resistant to erosion and chemical alteration, and they incorporate uranium into their crystal structure. The uranium decays into lead at a known rate, which allows scientists to determine precisely how old each zircon crystal is. Some of the oldest materials on Earth are zircons found in the Jack Hills of Western Australia. These crystals are over 4 billion years old, formed during the Hadean Eon, and they've survived everything Earth could throw at them across 4 billion years of geological time. But what's remarkable about these ancient zircons isn't just their age. It's what they tell us about the conditions under which they formed. Zircons form in molten rock, in magma deep underground, or in volcanic eruptions. But the chemical composition of these ancient zircons suggests something unexpected. They seem to have formed in the presence of liquid water. The ratio of isotopes within the crystals indicates that the magma from which they crystallized had interacted with water. This implies that Earth had liquid water, and therefore oceans, as early as 4.4 billion years ago, only about 200 million years after the planet formed. This discovery revolutionized our understanding of the Hadean. Instead of an entirely molten hellscape, we now think that Earth cooled relatively quickly. Within a few hundred million years, the surface had solidified enough to form a crust. Water vapor in the atmosphere condensed and fell as rain, torrential downpours that may have lasted for thousands of years, gradually filling the first oceans. The planet was still violent, still experiencing frequent impacts and volcanic eruptions, but it wasn't entirely uninhabitable. There were places, perhaps many places, where liquid water existed in stable pools and seas. This matters enormously for the story of life's origin. Life, as we know it, requires liquid water. Every biochemical reaction that sustains life takes place in water. Proteins fold in water. DNA replicates in water. Cells are bags of water with membranes around them. If Earth had liquid water 4.4 billion years ago, then the conditions for life could have existed almost from the very beginning, and there's evidence that they did. In rocks from Western Australia, scientists have found carbon isotopes in minerals that formed 4.1 billion years ago. These carbon isotopes show the characteristic signature of biological processes, the preference for lighter isotopes that living organisms display. This doesn't prove that life existed that early. The carbon signatures could potentially have formed through non-biological processes. But it's suggestive. It's consistent with the idea that life might have emerged very early in Earth's history. The Hadean atmosphere was nothing like the one we breathe today. There was virtually no oxygen. The sky would have looked different, perhaps more hazy, perhaps tinged with colors we don't associate with Earth's sky. The sun was about 30% dimmer than it is today, but Earth wasn't frozen because of the thick greenhouse atmosphere. Carbon dioxide and methane trapped heat, keeping the surface temperature above the freezing point of water despite the dimmer sun. This early atmosphere probably came from two sources. Some of it was primordial, captured from the solar nebula when Earth formed, mostly hydrogen and helium. But most of that light gas quickly escaped to space. The more important source was volcanic outgassing. As the planet's interior sorted itself out, with heavy iron sinking to form the core and the lighter rocks rising to form the mantle and crust, volcanoes released enormous quantities of gas from the interior. These gases, water vapor, carbon dioxide, nitrogen, and smaller amounts of methane, ammonia, hydrogen sulfide, and others, formed the secondary atmosphere that would persist for billions of years. The Hadean was also when Earth's magnetic field strengthened. Deep in the planet's core, liquid iron swirls and flows, creating electrical currents that generate a magnetic field. This field extends far beyond Earth's surface, creating the magnetosphere, an invisible shield that deflects charged particles from the sun. Without this magnetic field, the solar wind would gradually strip away Earth's atmosphere, as has happened on Mars. The magnetic field was crucial for maintaining an atmosphere capable of supporting life. By the end of the Hadean, around 4 billion years ago, Earth had transformed. The bombardment had slowed, the crust had stabilized. Continents, small ones at first, had begun to form through volcanic activity and the collision of crustal plates. The oceans were extensive, covering most of the planet's surface. The atmosphere was thick and reducing, rich in gases that could react to form organic molecules. It was still not a hospitable place by our standards. The air was unbreathable. The water was likely hot, perhaps close to boiling in many places. The sky was dominated by volcanic eruptions and lightning storms. Ultraviolet radiation from the sun, unfiltered by an ozone layer that wouldn't exist for billions of years, bombarded the surface. But it was a world where chemistry could happen, where molecules could form and react and recombine, where energy was abundant, from the sun, from lightning, from volcanic heat, from the energy released by chemical reactions themselves. It was a world on the edge, balanced between chaos and order, between sterility and the possibility of something more. It was in this world, during the late Hadean or the early Archean that followed, that life somehow began. The exact when and where remain uncertain. But the stage was set. The ingredients were present. All that was needed was the right combination, the right sequence of events, the right bit of chemistry in the right place at the right time. And somewhere on that ancient Earth, in some warm pool or deep-sea vent or mineral-rich tidal flat, it happened. Lifeless molecules crossed that mysterious boundary and became something that could be called alive. The geological processes of the Hadean were fundamentally different from those we observe today. Without continents and with a thinner crust, plate tectonics may have operated differently, if it operated at all. Some scientists suggest that the Hadean crust was more like a stagnant lid, with volcanism occurring through hotspots rather than at plate boundaries. Others propose that a primitive form of plate tectonics began very early, as soon as the crust cooled enough to crack and move. Evidence for these early geological processes is frustratingly scarce. The problem is that the rock cycle destroys its own history. Rocks formed in the Hadean have been melted, metamorphosed, eroded, and recycled countless times over 4 billion years. Only the hardiest minerals, like those zircon crystals, survive to tell the tale. Most of the Hadean is a blank page in Earth's geological record, but we can make inferences from what we do know. We know that Earth's interior was hotter in the Hadean. The planet had recently formed through the collision of smaller bodies, and all that kinetic energy had been converted to heat. Additionally, there were far more radioactive elements in Earth's interior, elements like uranium and thorium that decay over time, releasing heat. This meant more volcanic activity, more magma rising from the mantle, more heat flow from the interior to the surface. This heat had consequences for any emerging life. On one hand, high temperatures are generally inhospitable to life. Most organisms can't survive in boiling water, let alone the much hotter temperatures found near volcanic vents. But on the other hand, heat provides energy, and energy is necessary for the chemical reactions that underlie life. Some of Earth's most ancient organisms, based on genetic analyses, appear to have been thermophiles, heat-loving organisms that thrived in hot water. This suggests that high temperatures might not have been a barrier to early life, but rather a requirement. The oceans of the Hadean were probably different from modern oceans in another important way. They were likely more acidic. Carbon dioxide dissolves in water to form carbonic acid, and with the thick carbon dioxide atmosphere, the early oceans probably had a pH closer to that of vinegar than to modern seawater. This acidity would have implications for what kinds of chemistry could occur, what minerals would dissolve or precipitate, what kinds of molecular structures would be stable. Some scientists have suggested that life didn't begin in the ocean at all, but rather in freshwater environments on land. Shallow pools, perhaps in volcanic regions where geothermal heat kept the water warm, might have provided the right combination of concentration and chemistry for life to emerge. These pools would have gone through wet-dry cycles as water evaporated and was replenished by rain, and these cycles could have helped concentrate organic molecules and drive reactions that wouldn't occur in the more dilute environment of the ocean. Darwin's warm little pond might not have been far off after all. The transition from the Hadean to the Archean Eon, 4 billion years ago, wasn't marked by any single dramatic event. Rather, it represents a point where geologists agree that solid rocks begin to appear in the geological record. It's a practical boundary more than a natural one, but it does represent a time when Earth had fundamentally changed. The planet had cooled, the bombardment had slowed, stable continents were forming. The stage was set for the next act in Earth's story, and that act was the emergence of life itself. Before we can understand how life began, we need to understand what life is made of. Strip away all the complexity, all the diversity of living things, and you find that life is chemistry. Extraordinarily complex chemistry, to be sure, but chemistry nonetheless. And that chemistry is built on surprisingly simple foundations. Life, as we know it, is based on carbon. Every living thing on Earth, from the smallest bacterium to the largest whale, is made primarily of organic molecules, molecules with carbon atoms at their core. This isn't arbitrary. Carbon has unique properties that make it ideal for building the complex molecules that life requires. Carbon has 4 bonding sites, 4 places where it can attach to other atoms. This allows carbon atoms to link together in long chains, branching structures, and rings. Carbon can bond to hydrogen, oxygen, nitrogen, phosphorus, and sulfur, creating an almost limitless variety of molecular architectures. No other element can match carbon's versatility. Silicon comes closest, and some scientists have speculated about silicon-based life existing elsewhere in the universe. But silicon bonds are weaker and less flexible than carbon bonds. On Earth, at least, carbon is the foundation of life. The simplest organic molecules are the hydrocarbons, molecules made entirely of carbon and hydrogen. Methane, with 1 carbon atom and 4 hydrogen atoms, is the simplest. It was abundant in Earth's early atmosphere. But methane alone isn't enough for life. We need molecules with more complexity, with more varied properties. Life requires 4 major classes of organic molecules. First, there are amino acids, the building blocks of proteins. Proteins are the workhorses of the cell, carrying out most of the chemical reactions that sustain life, providing structure to cells, transporting molecules from place to place, and serving as messengers and regulators. There are 20 amino acids commonly used by life, and they can be linked together in any order to form chains called polypeptides. When these polypeptides fold into specific 3-dimensional shapes, they become proteins. Second, there are nucleotides, the building blocks of DNA and RNA. These molecules carry genetic information, the instructions for building and operating a living cell. DNA is the long-term storage medium, a stable molecule that can be copied accurately and passed from generation to generation. RNA is DNA's more versatile cousin, serving as a messenger that carries instructions from DNA to the protein-making machinery, and also as a catalyst that speeds up certain chemical reactions. Third, there are sugars, also called carbohydrates. These molecules provide energy and structure. Glucose is probably the most famous sugar, the primary fuel that powers most cells. But sugars can also link together to form longer chains like starch and cellulose. The ribose sugar in RNA and the deoxyribose sugar in DNA are part of the backbone of these genetic molecules. Fourth, there are lipids, fatty molecules that don't dissolve well in water. Lipids form the membranes that surround cells, creating a barrier between the inside and outside. Without membranes, without some way to concentrate molecules and separate them from the external environment, life as we know it couldn't exist. Lipids spontaneously form bubbles and sheets in water, a property that would prove crucial for the origin of life. These four classes of molecules, amino acids, nucleotides, sugars, and lipids, are the foundation of all known life. The question that puzzled scientists for centuries was how these complex organic molecules could have formed on the lifeless early Earth. Where did the building blocks come from? One possible answer came from space. When scientists analyzed meteorites that fell to Earth, they found organic molecules within them. The Murchison meteorite, which fell in Australia in 1969, contained more than 90 different amino acids, including many of the amino acids used by life on Earth. Other meteorites have been found to contain nucleobases, the molecular units that make up DNA and RNA, as well as sugars and other organic compounds. This discovery suggested that organic molecules might be common throughout the universe, forming in space through chemical reactions and delivered to planets by asteroids and comets. During Earth's early history, when bombardment was frequent, countless meteorites would have fallen to the surface, potentially bringing with them a rich inventory of prebiotic chemicals. Comets, which are mostly ice mixed with dust and organic compounds, could have delivered not only water to the early Earth, but also the raw materials for life. This idea is sometimes called panspermia, though that term is often used more specifically to refer to the hypothesis that life itself, rather than just organic molecules, might have originated elsewhere and been transported to Earth. Few scientists support the stronger version of panspermia, but many agree that extraterrestrial delivery of organic molecules probably played a role in providing the chemical feedstock for Earth's early chemistry. But could these molecules also form on Earth itself through chemical reactions in the atmosphere or oceans? That was the question that Alexander Oparin and John Haldane addressed in the 1920s. They proposed that Earth's early atmosphere, being reducing rather than oxidizing, could support reactions that created organic molecules from simpler gases. These molecules would rain down into the oceans, accumulating over time to create what Haldane called the primordial soup. The key insight was that the early Earth was chemically different from the modern Earth. Today's atmosphere is about 21% oxygen, and oxygen is a highly reactive gas. It oxidizes organic molecules, breaking them down. This is why organic matter decays, why iron rusts, why fires burn. In an oxygen-rich atmosphere, complex organic molecules would be unstable, they would react and break apart. But the early Earth had virtually no free oxygen. The atmosphere was reducing, dominated by gases like hydrogen, methane, and ammonia. In such an environment, organic molecules could form and persist. Given the right energy source, lightning or ultraviolet light from the sun, simple gases could react to form more complex molecules. Oparin and Haldane imagined the early oceans gradually becoming richer in organic compounds, a global soup in which increasingly complex chemistry could occur. This remained a theoretical idea until 1952, when Stanley Miller, a graduate student working with Harold Urey at the University of Chicago, decided to test it experimentally. His results were clear. Given gases thought to represent the early atmosphere and an energy source to drive reactions, organic molecules could indeed form spontaneously. The building blocks of life didn't require life itself to make them. They could arise through ordinary chemistry. Since Miller's time, scientists have discovered many other ways that organic molecules could have formed on the early Earth. Volcanic eruptions release gases that can react to form organic compounds. Minerals on rock surfaces can act as catalysts, speeding up reactions that would otherwise be too slow. Deep-sea hydrothermal vents create chemical gradients and provide energy that might drive organic synthesis. Even impacts, the very events that seem most destructive, might have contributed. When an asteroid or comet slams into the surface, the tremendous energy released can drive chemical reactions, creating organic molecules from the gases and minerals present. Recent experiments have shown that many of the molecular building blocks of life can form under a wide range of conditions. Amino acids form readily. Nucleobases, the components of DNA and RNA, can be synthesized from simpler precursors. Sugars like ribose can form through a process called the Formose reaction, where formaldehyde molecules link together. Lipids can form from simpler fatty acids. What's remarkable is that these syntheses don't require precisely tuned conditions. They're robust. They work in hot water and cool water, in acidic conditions and alkaline conditions, with different mixtures of starting materials. This suggests that wherever you have liquid water, basic chemical elements, and energy, you can probably make the molecular building blocks of life. But having the building blocks isn't the same as having life. A pile of bricks is not a house. A collection of amino acids and nucleotides is not a living cell. The challenge for scientists studying the origin of life has been to understand not just how the molecules formed, but how they organized themselves into something that could be called alive. This is where the problem becomes truly difficult. Modern cells are extraordinarily complex. A bacterial cell, which is about as simple as life gets, contains millions of molecules working in concert. There are thousands of different proteins, each one precisely folded to carry out a specific function. There's a genome, typically made of DNA, containing millions of base pairs, encoding the instructions for all those proteins. There's a membrane separating the cell from its environment, with proteins embedded in it to control what enters and leaves. There are ribosomes, the molecular machines that read RNA and build proteins. There are metabolic pathways, sequences of chemical reactions that extract energy from food molecules and use that energy to power the cell's activities. All of these components are interdependent. DNA needs proteins to replicate. Proteins need DNA to encode their sequences. Ribosomes need RNA and proteins. The cell membrane needs lipids and proteins. It's a chicken and egg problem on a massive scale. How could such a complex integrated system arise from simple chemistry? Scientists are convinced that it must have happened gradually through a series of steps, each one building on the last. The first proto-life forms wouldn't have had all the features of modern cells. They would have been much simpler, perhaps little more than self-replicating molecules enclosed in a membrane. Over time, through evolution and natural selection, these simple systems would have become more complex and more efficient. But identifying the specific steps, the actual pathway from chemistry to biology, remains one of the greatest challenges in science. We have many hypotheses, many possible scenarios, but we don't yet have a complete, detailed account of how it happened. That's the mystery we'll explore in the sections ahead, as we examine the experiments that have illuminated pieces of the puzzle, the environments that might have cradled early life, and the evidence from ancient rocks and modern genetics that constrains our theories. The building blocks were there. The chemistry was possible. What was needed was organization, selection, evolution. What was needed was a pathway from molecules to life. Understanding the chemistry of life also requires understanding the role of water. Water isn't just a passive medium in which reactions occur. It's an active participant in the chemistry of life, with properties that make it uniquely suited to support biological processes. Water is a polar molecule, meaning it has regions of positive and negative charge. This polarity allows water to dissolve many kinds of molecules, earning it the nickname, the universal solvent. But water doesn't dissolve everything. Lipids, for instance, don't mix well with water. This property is crucial for forming cell membranes. When lipid molecules are placed in water, they automatically arrange themselves into structures that minimize contact between their water-repelling tails and the water molecules. This self-organization creates bubbles and sheets without any guidance or instruction, a spontaneous emergence of structure from chaos. Water also stabilizes many biological molecules. The hydrogen bonds between water molecules and proteins help proteins fold into their correct three-dimensional shapes. The double helix of DNA is stabilized by water molecules surrounding it. Even the chemical reactions that build and break down biological molecules often involve water. Adding water to a bond breaks it apart in a process called hydrolysis. Removing water joins molecules together in a process called dehydration synthesis. Water's abundance on Earth made life possible, but the specific properties of water, its polarity, its ability to form hydrogen bonds, its role as both solvent and reactant, made life not just possible, but perhaps inevitable. Given water and the right chemical elements, life might be hard to prevent. This is one reason why the search for life elsewhere in the universe focuses so heavily on finding liquid water. Where there's water, there might be life. Of course, we can only speak confidently about carbon-based, water-dependent life because that's the only kind we know. It's possible that entirely different forms of life, based on different chemistry, exist elsewhere. But carbon and water seem like a good bet because they're abundant throughout the universe and because their chemical properties are so well suited to building complex, adaptable systems. Until we find an alternative, we look for life as we know it, life built from the same four classes of molecules, dissolved and organized by water, following the same basic principles of chemistry and physics that operate everywhere in the cosmos. In 1952, in a laboratory at the University of Chicago, a young graduate student named Stanley Miller performed an experiment that would change our understanding of life's origin and capture the imagination of the world. It was one of those rare moments in science when a simple idea, executed with elegant simplicity, produces results so dramatic and so clear that they instantly transform their field. Miller's experiment was designed to test the Oparin-Haldane hypothesis. If the early Earth had a reducing atmosphere, and if energy sources like lightning were common, could organic molecules form spontaneously from simple gases? Could the building blocks of life arise through nothing more than chemistry? The experiment itself was surprisingly simple. Miller built an apparatus from glass flasks and tubes. He filled it with water and gases, methane, ammonia, and hydrogen, along with water vapor. This mixture was meant to represent the early Earth's atmosphere as it was understood at the time. Then he applied energy to the system in the form of electrical sparks, simulating lightning strikes. The apparatus cycled continuously, with water evaporating from a heated flask, mixing with the gases, passing through the spark discharge, and then condensing back into a liquid that collected in a trap at the bottom. Miller let the experiment run for a week, monitoring the changes. Within a day or two, the liquid began to change color, first turning pink, then yellow, and eventually a murky brown. When Miller analyzed the brown liquid using paper chromatography, a technique that separates molecules based on their properties, he found something astonishing. The liquid contained amino acids. Not just one or two, but several of the amino acids used by living organisms, including glycine and alanine. He had created the building blocks of proteins from simple gases and electricity. The results were published in the journal Science in May of 1953, just 2 months after Watson and Crick announced the structure of DNA. Together, these discoveries marked a pivotal moment in biology. The structure of DNA showed how genetic information could be stored and copied. The Miller-Urey experiment, as it came to be known, showed how the molecules that make up living things could have formed on the primordial Earth. The experiment made headlines around the world. Here was experimental proof that life's building blocks didn't require divine intervention or mysterious vital forces. They could form through ordinary chemistry under conditions that likely existed on the early Earth. The experiment seemed to bridge the gap between non-life and life, showing that at least part of the pathway from chemicals to cells was accessible to scientific investigation. In the decades following Miller's initial experiment, he and other scientists performed many variations. They tried different gas mixtures, representing different theories about what the early atmosphere might have contained. They used different energy sources, not just electrical sparks, but also ultraviolet light, heat from volcanic sources, and shock waves simulating meteorite impacts. They varied the pH, the temperature, the pressure. What they found was that organic molecule formation is robust. Under a wide variety of conditions, as long as the atmosphere was reducing, amino acids and other organic compounds formed readily. The Miller-Urey experiment wasn't a lucky accident. It was demonstrating a fundamental principle. Given the right chemical ingredients and energy, organic synthesis occurs spontaneously. But as understanding of Earth's early atmosphere improved, a problem emerged. Evidence from ancient rocks and from studies of volcanic gases suggested that the early atmosphere probably wasn't as strongly reducing as Miller had assumed. Instead of being dominated by methane and ammonia, it more likely contained carbon dioxide and nitrogen, gases that are more chemically stable and less reactive. In such a weakly reducing or neutral atmosphere, the Miller-Urey experiment produced much lower yields of organic molecules. Amino acids still formed, but in smaller quantities. This led to decades of debate about whether the Miller-Urey experiment was relevant to the actual origin of life on Earth. Some scientists argued that it wasn't, that the experiment, while interesting, didn't reflect the real conditions of the early planet. Others pointed out that even with a neutral atmosphere, some organic molecules did form, and that other mechanisms, like delivery of organics by meteorites or synthesis near volcanic vents, could have supplemented atmospheric production. The debate took an interesting turn in 2008, after Miller's death. Scientists examined old vials from Miller's laboratory, samples from experiments he had performed but never published. One set of samples came from an experiment Miller had done in 1958 that simulated a volcanic plume with water, carbon dioxide, hydrogen sulfide, and other volcanic gases. When these samples were analyzed using modern, more sensitive techniques, they revealed a far richer mixture of organic compounds than Miller had detected with his 1950s-era equipment. The experiment had produced 23 different amino acids, including several that are used by life. Other unpublished experiments, analyzed decades later, showed similar results. Miller's original experiment had been more successful than he knew. His methods were right, even if some of the specifics about the atmosphere turned out to be different. The broader conclusion held. The building blocks of life could form through abiotic chemistry on the early Earth. Modern versions of the Miller-Urey experiment continue to refine our understanding. Recent work has shown that even with a weakly reducing atmosphere containing carbon dioxide and nitrogen, if you add iron minerals that would have been abundant on the early Earth, amino acid yields increase dramatically. The minerals act as catalysts and buffers, facilitating reactions that produce organic compounds. Other experiments have shown that wet-dry cycles, like those that would occur in tidal pools or around volcanic hot springs, can drive the formation of amino acids even more efficiently than a continuous aqueous environment. The Miller-Urey experiment also demonstrated something profound about the universe itself. The same chemical elements that were abundant on the early Earth, carbon, hydrogen, oxygen, nitrogen, are abundant throughout the cosmos. The same physical laws that allowed amino acids to form in Miller's apparatus operate everywhere. This suggests that the chemical building blocks of life might be common in the universe, forming wherever conditions permit, not just on Earth, but potentially on countless other worlds. We've since found evidence supporting this idea. Radio telescopes have detected complex organic molecules in interstellar space, in the clouds of gas and dust between stars. Spacecraft have analyzed the atmospheres of Saturn's moon Titan and found it rich in organic compounds, despite Titan being far too cold for life as we know it. Meteorites, as mentioned earlier, carry amino acids and other organic molecules. The universe appears to be a factory for producing the chemical ingredients of life. But the Miller-Urey experiment also revealed the limits of prebiotic chemistry. Yes, amino acids can form spontaneously, but amino acids are just the beginning. Life requires amino acids to link together in specific sequences to form proteins. It requires nucleotides to link together to form RNA or DNA. It requires these complex molecules to work together in a coordinated system. Miller's experiment created amino acids, but it didn't create proteins. It couldn't. Random chemical reactions don't create the highly specific sequences found in functional proteins. For that, you need some kind of template, some mechanism for selecting certain sequences over others, some form of heredity and evolution. The Miller-Urey experiment showed how you could get from simple gases to simple organic molecules. But the pathway from simple organic molecules to living cells remained mysterious. Nevertheless, The Miller-Urey experiment remains one of the landmarks of origin of life research. It transformed the field from speculation to experimentation. It demonstrated that at least part of the problem could be solved through chemistry. And it inspired generations of scientists to ask the next questions, to push further, to try to understand not just how the building blocks formed, but how they assembled into life itself. The experiment also changed how we think about life in the universe. If organic molecules form so readily, if the chemistry of life is just ordinary chemistry following ordinary physical laws, then perhaps life itself is more common than we imagined. Perhaps we're not alone in this vast cosmos. Perhaps wherever there's a planet with liquid water and the right chemical ingredients, life finds a way to begin. That's one of the most profound implications of Miller's simple apparatus of glass flasks and electrical sparks. It suggests that life might not be a cosmic accident, a one-time miracle that occurred only on Earth. It suggests that life might be, if not inevitable, then at least reasonably likely wherever conditions permit. And that transforms our understanding of our place in the universe. The Miller-Urey experiment also raised important questions about methodology in origin of life research. How do you design an experiment to test something that happened 4 billion years ago? What counts as evidence? How closely does a laboratory simulation need to match the actual early Earth for its results to be meaningful? These questions have no perfect answers, but they've led to increasingly sophisticated experimental approaches. Modern prebiotic chemistry experiments don't just mix gases and apply energy. They try to model specific environments that might have existed on the early Earth. They consider not just which molecules form, but which molecules persist, which can polymerize into larger structures, which might have the properties needed for self-replication or catalysis. Some experiments focus on mineral surfaces, testing whether clay minerals or metal sulfides could have acted as templates or catalysts, organizing organic molecules in ways that promote their interaction and polymerization. Other experiments explore cycles of wetting and drying, freezing and thawing, heating and cooling, testing how these rhythms might have driven chemical reactions forward. Still others investigate compartmentalization, showing how lipids can spontaneously form bubbles that could serve as protocells, concentrating molecules and creating chemical microenvironments. The Miller-Urey experiment also illustrates a key principle of experimental science. Even when the specific details are wrong, a well-designed experiment can still reveal fundamental truths. Miller's atmosphere wasn't quite right, but his broader conclusion that organic molecules can form through abiotic processes has stood the test of time. Science progresses not through perfect knowledge, but through successive approximations, each iteration bringing us closer to understanding reality. Today, when scientists conduct prebiotic chemistry experiments, they're building on Miller's foundation. They're using more sophisticated analytical techniques that can detect molecules present in tiny quantities. They're running computer simulations alongside laboratory experiments, modeling the chemistry at a molecular level to understand which reactions are likely and which are not. They're examining meteorites and analyzing data from space missions to other worlds, testing whether the chemistry they observe in the laboratory is consistent with what they observe in the cosmos. One surprising finding from these modern experiments is that amino acids might not have been the first important biological molecules to form. Some research suggests that RNA components, the nucleotides that make up genetic material, might have formed even more readily than amino acids under certain conditions. This has led to the RNA world hypothesis, which we'll explore in a later section, suggesting that the first self-replicating molecules might have been made of RNA rather than protein. Another important development has been the realization that the early Earth wasn't uniform. Different locations would have had different chemistries. A volcanic vent would have different conditions than a tidal pool, which would differ from a deep ocean environment. The Miller-Urey experiment simulated one possible environment, but the early Earth had many environments, each with its own characteristic chemistry. Perhaps different building blocks formed in different places, and the origin of life required bringing together molecules from multiple sources. The legacy of the Miller-Urey experiment extends beyond its specific scientific findings. It demonstrated that the origin of life is a legitimate scientific question, amenable to experimental investigation. It showed that you don't need to invoke miracles or unknowable processes. You can test hypotheses, gather data, refine theories. The origin of life is a mystery, yes, but it's a mystery that science can address. And 60 years later, scientists are still conducting variants of Miller's experiment, still exploring prebiotic chemistry, still working to understand how life began. The questions have grown more sophisticated, the techniques more advanced, but the fundamental goal remains the same: to understand how chemistry became biology on the young Earth. While Stanley Miller was simulating Earth's early atmosphere in a laboratory, dramatic discoveries were being made deep beneath the ocean surface. These discoveries would suggest an entirely different environment for life's origin, an environment that seems, at first glance, utterly inhospitable, but which might actually be the perfect cradle for life. In 1977, a team of scientists aboard the research submarine Alvin were exploring the Galapagos Rift, a volcanic crack in the ocean floor in the eastern Pacific. They were searching for hydrothermal vents, places where seawater seeps into cracks in the oceanic crust, gets heated by hot rock deep below, and then erupts back into the ocean. Such vents had been predicted to exist, but no one had seen one directly. What they found exceeded their expectations. The first vent they encountered was releasing water heated to over 300 degrees Celsius. At the crushing pressures of the deep ocean, this superheated water didn't boil. Instead, it jetted upward in a plume that looked like smoke, dark with dissolved minerals, mainly iron and sulfur compounds, that precipitated out when the hot water mixed with the frigid surrounding seawater. The scientists called them black smokers because of the dark, billowing clouds they produced. But what truly astonished the researchers wasn't the vents themselves. It was what lived around them. Despite being 2.5 kilometers below the surface, in total darkness, under pressure that would crush an unprotected human instantly, in waters alternately scalding and freezing, the vents were surrounded by thriving ecosystems. There were giant tube worms, some over 2 meters long, clustered around the vents. There were shrimp, crabs, clams, and fish, all adapted to this extreme environment. It was an entire biological community in one of the most unlikely places imaginable. For decades, scientists had assumed that all life on Earth ultimately depended on the sun. Plants and algae use photosynthesis to convert sunlight into chemical energy. Herbivores eat the plants. Carnivores eat the herbivores. Even in the deep ocean, life was thought to depend on organic matter drifting down from the sunlit surface waters. But here, in the eternal darkness of the deep sea, life thrived without any connection to photosynthesis. The key turned out to be bacteria. Chemosynthetic bacteria living near the vents harness the chemical energy released when hydrogen sulfide, dissolved in the vent water, reacts with oxygen in the seawater. They use this energy to convert carbon dioxide into organic molecules, just as plants use sunlight to do the same. These bacteria form the base of the vent ecosystem, supporting all the larger organisms. The giant tube worms, for instance, have no mouth or digestive system. Instead, they have a specialized organ filled with chemosynthetic bacteria. The worm's bright red plume absorbs hydrogen sulfide and oxygen from the water and transports them to the bacteria. The bacteria produce organic compounds that nourish the worm. It's a perfect symbiosis, and it works without a single photon of sunlight. The discovery of hydrothermal vent ecosystems revolutionized biology. It showed that life could thrive in conditions previously thought impossible. It demonstrated that chemosynthesis could support complex ecosystems, and it suggested a radical idea. If life could exist at hydrothermal vents today, perhaps life originated at hydrothermal vents 4 billion years ago. The idea has considerable appeal. Hydrothermal vents provide several things that life needs. They provide energy in the form of chemical disequilibrium. Hot, reducing fluids from deep in the earth mix with cold, oxidizing seawater, creating conditions where reactions can occur that release energy. They provide a steady supply of raw materials, minerals, and dissolved gases that could serve as building blocks for organic molecules. They provide concentration mechanisms. As minerals precipitate from the hot water, they can trap and concentrate organic molecules. And they provide protection. In the late Hadean, when the Earth's surface was being bombarded by asteroids, the deep ocean floor might have been one of the few places where life could survive. There are actually two main types of hydrothermal vents, and they might represent two different potential environments for life's origin. Black smokers, the first type discovered, are hot and acidic. They form at mid-ocean ridges where tectonic plates are pulling apart and magma rises close to the surface. The water from black smokers can exceed 400 degrees Celsius and has a pH around 3, as acidic as vinegar. The minerals dissolved in this water are mainly iron and sulfur compounds, giving the plumes their characteristic black color. The other type, called white smokers or alkaline vents, was discovered later. These vents are cooler, usually around 40 to 90 degrees Celsius, and alkaline, with a pH around 10 or 11. They form not at spreading ridges, but at places where seawater reacts with rocks in the ocean crust, particularly rocks rich in olivine and pyroxene. This reaction, called serpentinization, produces hydrogen gas and creates alkaline, mineral-rich fluids. When these fluids emerge from the seafloor, they precipitate minerals like calcium carbonate and magnesium hydroxide, forming white or pale-colored chimney structures. The most famous example of alkaline vents is a site called the Lost City, discovered in the year 2000 on the floor of the Atlantic Ocean. The vents at the Lost City form spectacular white towers, some over 60 meters tall, built from layers of carbonate minerals deposited over thousands of years. The fluids emerging from these vents support dense microbial communities, and the temperature and pH are much more compatible with life as we know it than the extreme conditions at black smokers. Many origin of life researchers now favor alkaline vents over black smokers as the most likely birthplace of life. The chemistry is less harsh, the temperatures more moderate, and alkaline vents provide something else that might be crucial: natural chemical gradients. The pH difference between the alkaline vent fluid, around 10 or 11, and the acidic seawater of the early ocean, probably around 5 or 6, creates a proton gradient across the thin mineral walls of the vent chimneys. This matters because modern cells use proton gradients to generate energy. In a process called chemiosmosis, cells pump protons across a membrane, creating a difference in concentration and electrical charge. The protons then flow back through special proteins that harness this flow to make ATP, the energy currency of the cell. This process is so fundamental to life that it's found in bacteria, archaea, and eukaryotes. Every living cell uses proton gradients to store and release energy. The fact that alkaline hydrothermal vents naturally provide proton gradients similar to those used by cells is striking. Some scientists, particularly Nick Lane at University College London and Michael Russell at NASA's Jet Propulsion Laboratory, have proposed that life might have originated in the porous rocks of alkaline vent chimneys. These rocks have countless tiny compartments, natural protocells bounded by thin mineral walls. The proton gradient across these walls could have powered the first chemical reactions that led to life, the same way proton gradients power modern cells. There's experimental support for this idea. Researchers have created artificial vent chimneys in the laboratory by injecting alkaline fluids into acidic water. These artificial chimneys spontaneously develop the same kinds of porous, compartmentalized structures seen in natural vents. When supplied with hydrogen, carbon dioxide, and simple mineral catalysts, these artificial vents can drive the formation of organic molecules, including some of the building blocks of nucleotides and amino acids. Other experiments have shown that metal sulfide minerals, common in both types of hydrothermal vents, can act as catalysts for organic synthesis. Iron-sulfur compounds, in particular, seem to have been important in the origin of life. Many modern enzymes have iron-sulfur clusters at their active sites, the places where chemistry happens. These clusters might be molecular fossils, remnants of a time when all of life's chemistry was catalyzed by inorganic iron-sulfur minerals, rather than by protein enzymes. Hydrothermal vents also solve a problem that plagues some origin of life theories: dilution. In the open ocean, organic molecules would be spread very thin, making it unlikely they'd encounter each other frequently enough to react and form larger structures. But in the confined spaces of vent chimneys, in the tiny pores and channels within the rock, molecules would be concentrated. They'd be held close to mineral surfaces that could catalyze reactions. The flowing vent fluids would continuously bring in fresh reactants while carrying away products, driving reactions forward. There are challenges with the vent hypothesis, of course. The fluctuating temperatures and the harsh chemical environment might break down complex molecules as fast as they form. The high salt content of seawater can interfere with some reactions that seem important for life. And it's not clear how life could have transitioned from being embedded in rock to being enclosed in membranes, from being dependent on geological energy sources to generating its own energy through biochemistry. But these challenges aren't insurmountable. They're puzzles to be solved through more research, more experiments, more theoretical work. And the appeal of the vent hypothesis remains strong. Vents provide energy, raw materials, compartments, and concentration mechanisms. They provide a plausible environment where the chemistry of life could have gotten started. Moreover, hydrothermal vents aren't unique to Earth. Scientists believe they exist on other worlds in our solar system. Jupiter's moon Europa and Saturn's moon Enceladus both have subsurface oceans beneath thick layers of ice, and both show evidence of hydrothermal activity. If life could originate at hydrothermal vents, then these moons might be habitable, despite receiving virtually no sunlight. The search for life beyond Earth increasingly focuses on these icy ocean worlds, and understanding how life might begin at vents is crucial for knowing where and how to look. The discovery of vent ecosystems also transformed our understanding of life's resilience and adaptability. Organisms called extremophiles thrive in boiling water, in acid, in saturated salt solutions, in high radiation, in the deepest parts of the ocean. Life is far tougher and more versatile than we once thought. And if life can thrive in such extreme environments today, perhaps it began in an extreme environment, under conditions that would seem utterly hostile by our surface-dwelling standards. Deep-sea vents remind us that life doesn't require comfortable, moderate conditions. It requires energy, raw materials, and liquid water. Vents provide all three. Whether they were actually the site of life's origin remains uncertain, but they represent a compelling possibility, an environment where the first self-replicating molecules might have emerged from the interplay of geology and chemistry, energy and entropy, chaos and order. Here's a puzzle that has perplexed origin of life researchers for decades. Modern life uses DNA to store genetic information, RNA to carry that information from the nucleus to the cellular machinery, and proteins to do almost everything else, from catalyzing chemical reactions to providing structure to the cell. But DNA can't replicate without proteins, and proteins can't be made without instructions from DNA. It's the ultimate chicken and egg problem. Which came first? The answer, many scientists now believe, is neither. Or rather, both functions, information storage and catalysis, were once carried out by a single type of molecule, RNA. This idea is called the RNA world hypothesis, and it's one of the most important concepts in modern origin of life research. To understand why scientists think there was once an RNA world, we need to understand what RNA is and what it can do. RNA, or ribonucleic acid, is chemically similar to DNA. Both are nucleic acids, long polymers made of individual units called nucleotides. Each nucleotide consists of 3 parts: a sugar molecule, a phosphate group, and a nitrogen-containing base. In DNA, the sugar is deoxyribose. In RNA, it's ribose, which has 1 extra oxygen atom. This small difference makes RNA more chemically reactive than DNA, which matters in ways we'll explore. The bases in DNA are adenine, guanine, cytosine, and thymine. RNA uses the same bases, except it replaces thymine with uracil, a similar base that pairs with adenine. These bases pair up in predictable ways: adenine with thymine or uracil, and guanine with cytosine. This base pairing is what allows DNA and RNA to store information. The sequence of bases along the molecule encodes genetic information, much like letters encode words. For most of the 20th century, scientists thought DNA and RNA served only to store and transmit information, while proteins did all the chemistry. But in the 1980s, 2 independent discoveries shattered this assumption. Thomas Sech, studying a single-celled organism called tetrahymena, found that an RNA molecule could splice itself, cutting out a section of itself and rejoining the ends without any protein enzymes to help. Around the same time, Sidney Altman discovered that RNase-P, an enzyme involved in processing RNA, consisted of both RNA and protein, and that the RNA part was the one actually doing the chemistry. These RNA molecules that could catalyze chemical reactions were called ribozymes. For discovering them, Sech and Altman shared the 1989 Nobel Prize in Chemistry. Their discoveries proved that RNA wasn't just a passive information carrier. It could be an active catalyst, performing the kinds of chemical reactions that everyone had thought required proteins. This revelation transformed thinking about the origin of life. If RNA could both store information and catalyze reactions, then perhaps early life was based entirely on RNA. Perhaps the first self-replicating molecules were RNA molecules that could make copies of themselves. Perhaps DNA and proteins came later, taking over the roles of information storage and catalysis respectively, because they could do those jobs more efficiently. This is the essence of the RNA world hypothesis. It proposes that there was a stage in the evolution of life when RNA did everything. RNA genomes stored genetic information. RNA ribozymes catalyzed the chemical reactions needed to copy those genomes and to carry out primitive metabolism. RNA was both chicken and egg. The appeal of this idea is enormous. It solves the interdependence problem. You don't need DNA to make proteins and proteins to make DNA if RNA can handle both functions. It provides a plausible intermediate stage between simple organic molecules and the complex DNA protein machinery of modern cells, and it's supported by molecular fossils within modern cells. Consider the ribosome, the molecular machine that makes proteins. Ribosomes are made of both RNA and protein. For years, scientists assumed the protein components were doing the actual work of linking amino acids together. But high-resolution structural studies in the late 1990s and early 2000s revealed something surprising. The active site of the ribosome, the place where amino acids are joined to form proteins, is made entirely of RNA. The proteins provide structural support, but the RNA does the chemistry. This suggests that the ribosome is an ancient structure, a molecular fossil from the RNA world. The ribosome might have started as an all-RNA machine, with proteins added later to improve its function. The fact that modern ribosomes still use RNA to catalyze protein synthesis hints at their evolutionary history, like a modern car whose engine still contains parts designed for an earlier era. Other examples of ribozymes exist in modern cells. RNA molecules catalyze the splicing of other RNA molecules. They catalyze reactions involved in making more RNA. These activities make sense if RNA once had to do everything on its own, before proteins took over most catalytic roles. But the RNA world hypothesis faces serious challenges. The first is the origin of RNA itself. Ribonucleotides, the building blocks of RNA, are complex molecules. They consist of a sugar, a base, and a phosphate group, all precisely arranged. Making ribonucleotides from simpler molecules turns out to be difficult. The sugar, ribose, can form through the 4-Mos reaction, but this reaction produces many different sugars, and it's not clear how nature would have selected ribose specifically. Attaching a base to the sugar is chemically tricky, and linking ribonucleotides together to form RNA polymers is challenging without enzymes to help. Scientists have worked for decades to find plausible prebiotic routes to RNA, with mixed success. Some progress has been made. In 2009, John Sutherland and his colleagues at the University of Cambridge showed that ribonucleotides could be synthesized from simpler molecules through a series of reactions that seem plausible for the early Earth. Their synthesis didn't work the way biochemists had expected, building up the sugar, base, and phosphate separately and then joining them. Instead, the molecules assembled in an unexpected sequence, with intermediates that formed and reacted in ways that biochemists hadn't anticipated. This work suggested that nature's path to RNA might have been different from the paths that modern cells use, and that by thinking creatively about chemistry rather than biochemistry, Scientists might be able to reconstruct the prebiotic synthesis of RNA building blocks. Still, the synthesis required carefully controlled conditions and pure starting materials, raising questions about whether it could realistically have occurred on the early Earth. Another challenge for the RNA world is that RNA is unstable. It's much more prone to degradation than DNA, breaking down in the presence of water, heat, or alkaline conditions. This fragility might seem like a problem for a molecule that was supposed to serve as the genetic material for early life, but it might also have been an advantage. The chemical reactivity that makes RNA unstable is the same reactivity that allows it to catalyze reactions. RNA walks a tightrope between stability and reactivity, and that balance might have been necessary for the emergence of life. Modern cells protect their RNA from degradation through various modifications, adding methyl groups and other chemical tags that stabilize the molecule. Perhaps early RNA molecules evolved similar protective modifications, allowing them to persist long enough to replicate. A third challenge is that ribozymes, while versatile, have limitations. They can catalyze many reactions, but not as efficiently as protein enzymes. The most important ribozyme for the RNA world would be an RNA polymerase, an RNA molecule that could copy other RNA molecules. Scientists have spent years trying to evolve such a molecule in the laboratory. The best RNA polymerase ribozymes created so far can copy RNA sequences about 95 nucleotides long, which is impressive, but falls short of what would be needed for a fully self-replicating system. Such a system would need an RNA polymerase ribozyme that could copy itself, meaning it would need to copy an RNA molecule at least as long as the polymerase itself. That's a formidable challenge, and it hasn't yet been achieved in the laboratory. Some scientists suggest that the RNA world might not have been populated by individual self-replicating RNA molecules, but rather by communities of RNA molecules that collectively sustained their own replication. One RNA molecule might copy another, which copies a third, which copies the first. This networked approach would avoid the need for any single RNA molecule to replicate itself directly. Evolution would act on the network as a whole, favoring combinations of RNA molecules that together could sustain the system. Others have proposed that RNA wasn't actually the first genetic molecule. Perhaps there was a pre-RNA world based on simpler nucleic acids that were easier to synthesize but less efficient. Candidates include PNA, peptide nucleic acid, which has a protein-like backbone instead of the sugar phosphate backbone of RNA, or TNA, 3-O's nucleic acid, which uses a simpler 4-carbon sugar instead of ribose's 5 carbons. These alternative nucleic acids might have preceded RNA, with RNA later taking over because of its superior properties. Despite these challenges, the RNA world hypothesis remains the leading theory for an intermediate stage in the origin of life. It's supported by the existence of ribozymes in modern cells, by the central role of RNA in protein synthesis, and by experimental demonstrations that RNA can catalyze a wide range of reactions. It provides a conceptual bridge between the purely chemical world and the DNA protein world of modern life. If there was an RNA world, it probably didn't last forever. RNA has advantages, but DNA and proteins have their own advantages. DNA is more stable, better for long-term information storage. Proteins are more versatile catalysts with a wider range of chemical capabilities. Once cells evolved the machinery to make DNA and proteins, these molecules gradually took over the roles that RNA had been playing. but RNA retained its central position in protein synthesis, a reminder of its ancient importance. The RNA world hypothesis also has implications for the search for life elsewhere. If life elsewhere follows a similar chemical path as Earth life, then we might expect to find RNA or something like it. The ubiquity of RNA in Earth life, its presence in all three domains, bacteria, archaea, and eukaryotes, suggests that RNA was present in Luca, the last universal common ancestor. This means RNA predates the diversification of life into these three great branches, placing it very early in life's history. The story of the RNA world is still being written. Scientists continue to search for more efficient ribozymes, better prebiotic syntheses of RNA components, and evidence in modern biochemistry for ancient RNA functions. Each new discovery adds a piece to the puzzle, helping us understand how a world of self-replicating RNA molecules might have given rise to the more complex biochemistry we see today. And as we peer backward through time, trying to understand how life began, the RNA world stands as a milestone on that journey, a plausible intermediate stage between lifeless chemistry and living cells, a time when a single versatile molecule carried within itself both the instructions for life and the tools to execute those instructions. We've explored the chemical building blocks, the environments where they might have formed, and the possibility that RNA served as an intermediate stage between simple chemistry and complex life. But there's still a vast gulf to cross. Having self-replicating RNA molecules, if they existed, is not the same as having a living cell. The transition from chemistry to biology, from molecules to life, remains one of the most profound mysteries in science. What makes something alive? It's a harder question than it might seem. Biologists have proposed many definitions, but none is perfect. Is a virus alive? It can reproduce, but only by hijacking a cell's machinery. Is a seed alive? It's dormant, not metabolizing, yet it can grow into a plant. Is a prion, a misfolded protein that causes diseases like mad cow disease, alive? It can replicate by converting other proteins to its shape, but it has no genetic material, no metabolism. Most definitions of life include several key features. Life must be able to reproduce, making copies of itself. It must have some form of heredity, passing information from parent to offspring. It must have metabolism, carrying out chemical reactions to extract energy from the environment. It must maintain itself, keeping its internal organization distinct from the external environment. And it must be capable of evolution, changing over time in response to environmental pressures. A living cell has all these properties. It can reproduce through cell division. It has a genome, DNA or RNA, that it copies and passes to daughter cells. It has metabolism, breaking down nutrients and using the energy to power its activities. It has a membrane that separates it from the environment, and populations of cells evolve, with natural selection favoring cells that reproduce more efficiently. But how do you get from simple organic molecules to something that meets all these criteria? This is the problem of the Great Transition, and solving it requires understanding several key innovations that early life must have achieved. First, there's the problem of replication. Molecules don't naturally copy themselves. In modern cells, DNA replication requires dozens of different protein enzymes working in a coordinated sequence. RNA replication is simpler, but it still requires specific catalysts. How could the first replicating molecules have reproduced without the complex machinery that modern cells use? The answer probably involves simple RNA, or pre-RNA molecules that could act as templates. If you have a polymer of nucleotides, and if free nucleotides in solution can pair up with the bases on that polymer, then you have the beginning of replication. The free nucleotides, guided by base pairing, line up along the template. If they can then link together, perhaps catalyzed by the template itself or by minerals, you get a complementary copy. This complementary copy can then serve as a template to make another copy, which will have the same sequence as the original. It's not perfect replication, and it wouldn't be very accurate, but it's a start. Evolution can work with imperfect replication as long as the copies are similar enough to the original that information is preserved across generations. Early replication was probably error-prone, but in some ways, that's good. Errors create variation, and variation is the raw material for evolution. Some variants might replicate faster or more accurately. Some might be more stable or better able to catalyze useful reactions. Over time, natural selection would favor these improved variants, leading to more sophisticated replication mechanisms. Second, there's the problem of compartmentalization. Modern cells are enclosed in membranes that separate their contents from the external environment. These membranes are made of lipids, fatty molecules that spontaneously form two-layered sheets in water. The lipid bilayer is semi-permeable, meaning it lets some molecules pass through while blocking others. This allows the cell to control its internal chemistry, accumulating useful molecules and excluding harmful ones. How did the first compartments form? Remarkably, this might have been the easiest problem to solve. Lipids spontaneously self-assemble into vesicles, bubble-like structures, when placed in water. This happens without any guidance or instruction. It's just physics and chemistry. The hydrophobic tails of lipid molecules avoid water, while the hydrophilic heads interact with it. The most stable configuration is a bilayer with the tails facing inward and the heads facing outward, forming a closed sphere. Scientists have created vesicles in the laboratory from simple fatty acids, lipids that could plausibly have existed on the early Earth. These vesicles can grow by absorbing more lipids from the surrounding water. They can divide when agitated, and they can encapsulate molecules, trapping them inside. If you add self-replicating RNA to a solution with lipids, some of the RNA gets trapped inside vesicles as they form. Now you have a protocell, a membrane-bound compartment containing genetic material. These protocells aren't alive, not yet, but they have some lifelike properties. They can compete for resources. Vesicles with more internal molecules grow faster by absorbing more lipids. If a vesicle contains RNA that catalyzes useful reactions, it might produce more nucleotides, allowing its RNA to replicate more. The vesicle could grow and divide, passing its RNA to daughter vesicles. This provides a mechanism for evolution to act. Vesicles with better RNA that replicates more efficiently or catalyzes more useful reactions, will outcompete vesicles with inferior RNA. Over time, the population of protocells would become more sophisticated, developing more complex chemistry and more efficient replication. Third, there's the problem of metabolism. Modern cells have intricate metabolic networks, sequences of chemical reactions that extract energy from food and use that energy to power the cell's activities. These pathways are catalyzed by dozens or hundreds of different enzymes, each one precisely evolved to carry out a specific reaction. How did metabolism begin? One idea is that it started with simple chemical reactions that released energy and could be catalyzed by minerals or by simple ribozymes. For example, In modern metabolism, many reactions involve iron-sulfur clusters. These clusters might be molecular fossils from a time when simple iron-sulfur minerals catalyzed the first metabolic reactions. One particularly interesting metabolic pathway is the reverse Krebs cycle, also called the reductive citric acid cycle. This pathway fixes carbon dioxide, converting it into organic molecules, and it can operate in a cycle that continually regenerates its starting materials. Some scientists have shown that this cycle, or something like it, can proceed non-enzymatically, catalyzed by simple iron and nickel sulfide minerals under conditions similar to those at hydrothermal vents. If the reverse Krebs cycle, or a similar pathway, operated in protocells at deep-sea vents, it could have provided both organic building blocks and energy. The protocells would be chemosynthetic, like modern vent bacteria, using chemical energy from the vent fluids rather than light from the sun. Fourth, there's the problem of the genetic code. Modern life uses a specific mapping between sequences of 3 DNA bases, called codons, and the 20 amino acids used in proteins. This code is nearly universal across all life, suggesting it was established very early and has been preserved with minor variations ever since. But why this particular code? Why these 20 amino acids? And how did the code originate in the first place? There's no obvious chemical reason why a particular codon should correspond to a particular amino acid. The mapping seems arbitrary, which suggests it was established through evolution rather than chemistry. One hypothesis is that the code started simple and expanded over time. Perhaps early life used only a few amino acids, and the code gradually added more as cells evolved the ability to synthesize them. Another idea is that the code has some optimization properties. It's somewhat error-resistant, meaning that mutations often change one amino acid to another with similar properties, minimizing the damage from copying errors. This suggests the code was shaped by natural selection. The origin of the genetic code remains one of the deepest mysteries in biology. Solving it would tell us a great deal about how the molecular machinery of life arose. Fifth, there's the transition from RNA to DNA. If there was an RNA world, how and why did DNA take over the role of genetic material? DNA is chemically more stable than RNA, which makes it better for long-term information storage. But making DNA requires converting ribonucleotides to deoxyribonucleotides, removing one oxygen atom. This is done by an enzyme called ribonucleotide reductase. The presence of this enzyme in all modern cells suggests that the transition from RNA to DNA occurred before LUCA, before the diversification of life into bacteria, archaea, and eukaryotes. Once DNA evolved, it gradually displaced RNA as the genetic material, though RNA retained its roles in protein synthesis and gene regulation. All of these transitions, from non-replicating molecules to replicating ones, from free-floating molecules to compartmentalized protocells, from simple chemistry to organized metabolism, from an arbitrary collection of molecules to an evolving system with heredity, represent steps on the path from chemistry to life. We don't know the exact sequence of events, which transitions occurred first, or how they were connected. But each one is necessary, and each one has plausible chemical mechanisms. The Great Transition wasn't a single event. It was a process, probably spanning millions of years, with countless dead ends and false starts. It happened not through any single brilliant innovation, but through the gradual accumulation of small improvements, each one building on the last, each one subject to selection and refinement. And somewhere along this path chemistry became biology. Molecules organized themselves into systems complex enough, integrated enough, adaptable enough that they could be called alive. The exact moment when this happened, the exact point at which non-life became life, may be impossible to identify. It might not even be a meaningful question. But the fact that it happened, that our planet gave rise to life, tells us something profound about the universe we inhabit. The transition from chemistry to biology also required solving informational problems, not just chemical ones. Modern organisms have genomes containing millions or billions of base pairs, encoding thousands of genes. Early life couldn't have had genomes that large. The first replicating molecules were probably short, perhaps a few hundred nucleotides at most. But even short RNA molecules can contain meaningful information if that information is expressed through their effect on chemistry. Consider a ribozyme that catalyzes its own replication. The sequence of bases in that ribozyme determines how it folds, which determines how it functions. This sequence is information, even if it's not a gene in the modern sense. And because this information affects how efficiently the molecule replicates, it's subject to natural selection. Better sequences that fold into more efficient catalysts, will outcompete worse sequences. This is the beginning of evolution at the molecular level. It doesn't require cells or complex organisms. It requires only molecules that can replicate and that have some influence on their own replication rate. Given that minimal system, evolution can proceed, refining the molecules and, over time, creating increasing complexity. The information content of early life probably grew gradually. Short sequences that performed useful functions could be linked together, creating longer molecules with more sophisticated capabilities. Mistakes in copying could occasionally create new functional sequences, and natural selection would preserve the innovations that worked while discarding those that didn't. If we could travel backward through evolution, following the branches of the tree of life toward its root, we would eventually arrive at a single organism from which all modern life descends. Scientists call this organism LUCA, the last universal common ancestor. It's not the first living thing. That honor belongs to whatever proto-organisms evolved from chemistry billions of years earlier. But LUCA is the last organism that was ancestral to everything alive today, from bacteria to blue whales, from sequoia trees to humans. For decades, scientists have tried to reconstruct what LUCA was like by looking for features shared by all living things. If all organisms have a particular gene or use a particular biochemical pathway, the logic goes, then LUCA probably had that gene or used that pathway too. By cataloging these universal features, scientists can paint a portrait of our earliest ancestor. Recent studies have revolutionized our understanding of LUCA. In 2024, an international team of researchers published an analysis that combined genomics, geology, and molecular clock estimates to determine when LUCA lived and what it was like. Their conclusion was startling. LUCA lived approximately 4.2 billion years ago, only about 400 million years after Earth formed, during the late Hadean or early Archean eon. This date is earlier than previous estimates, and it has profound implications. It means life emerged very quickly on Earth, almost as soon as conditions permitted. It suggests that the origin of life might not be as difficult or improbable as it sometimes seems. Given the right conditions, life might arise relatively rapidly. So what was LUCA like? The researchers analyzed thousands of genes across hundreds of modern species, looking for those that could be traced back to LUCA through evolutionary relationships. They found that Luca had a surprisingly large genome, encoding about 2,600 proteins. This is comparable to some modern bacteria, suggesting that Luca was already a fairly complex organism, not the simple protocell we might expect at life's very beginning. Luca was a prokaryote, meaning it lacked the membrane-bound nucleus and organelles found in eukaryotic cells like those in plants, animals, and fungi. It was a single-celled organism, probably similar in basic structure to modern bacteria or archaea. In fact, Luca is the organism from which both bacteria and archaea descended, representing the split that would eventually lead to two of the three great domains of life. Luca was anaerobic, meaning it didn't require oxygen, and probably couldn't tolerate it. This makes sense because the early Earth's atmosphere had virtually no free oxygen. Oxygen only became abundant about 2 billion years ago, when photosynthetic bacteria began producing it in large quantities in what's called the Great Oxygenation Event. For the first 2 billion years of life on Earth, organisms lived in an oxygen-free world. Luca obtained energy through chemosynthesis rather than photosynthesis. It used chemical reactions, likely involving hydrogen gas and carbon dioxide, to produce organic molecules and ATP. The specific metabolic pathway it used was probably the Wood-Ljungdahl pathway, a method of carbon fixation that's still used by some modern anaerobic bacteria and archaea. This pathway is particularly interesting because it's thought to be one of the most ancient forms of metabolism, possibly representing the way the first living cells obtained energy. The fact that Luca used the Wood-Ljungdahl pathway strongly suggests it lived near hydrothermal vents. This pathway requires hydrogen gas and carbon dioxide, both of which are abundant in vent environments. It works best at high temperatures and in the presence of iron-sulfur minerals, conditions characteristic of vents. Combined with the genetic evidence, this points to Luca as a vent-dwelling organism, thriving in the hot, mineral-rich waters near underwater volcanoes. Luca had DNA as its genetic material and used the same genetic code that virtually all modern organisms use. It had ribosomes, the molecular machines that synthesize proteins, though these were probably simpler than modern ribosomes. It had a cell membrane made of lipids, though the exact composition of this membrane remains uncertain. Interestingly, bacteria and archaea use different types of lipids in their membranes, suggesting that Luca's membrane might have been different from either, or that the membrane composition evolved independently in the two lineages after they split. One of the most intriguing findings from the 2024 study was that Luca had a rudimentary immune system. Genes related to the CRISPR-Cas system, which modern bacteria and archaea use to defend against viruses, appear to have been present in Luca. This is remarkable because it implies that viruses existed 4.2 billion years ago, and that Luca was already engaged in an evolutionary arms race with them. The presence of viruses so early in life's history suggests that they may have played a role in evolution from the very beginning. Viruses transfer genes between organisms, contributing to the exchange of genetic information that drives evolutionary innovation. They also exert selective pressure, favoring organisms that can defend against them. This virus-host interaction might have been a major driver of biological complexity even in Earth's earliest ecosystems. Luca probably wasn't alone. The analysis suggests it was part of an ecosystem, living alongside other microorganisms. Some of these other organisms might have been methanogens, microbes that produce methane gas as a byproduct of their metabolism. The waste products from Luca's metabolism could have served as nutrients for methanogens, which in turn would produce methane that could be used by other organisms. This recycling of materials, with one organism's waste becoming another's food, is characteristic of ecosystems, and it appears this principle was operating even in the earliest stages of life on Earth. It's important to understand what Luca was not. Luca was not the first living thing. Many generations of organisms must have lived and evolved before Luca. Luca represents a bottleneck, a point through which all modern life passed, but there were almost certainly other lineages that didn't make it, organisms that went extinct without leaving descendants in the modern world. Luca is called the last universal common ancestor for this reason. It's the last organism that was common to all modern life. There might have been earlier organisms ancestral to Luca, perhaps representing a proto-bacterial or proto-archaeal state, but those ancestors didn't give rise to all modern life, only to the lineage that led to Luca. The evolutionary tree of life has three main branches: bacteria, archaea, and eukarya. For many years, scientists debated how these three domains were related. Were they all equally ancient? Three separate lineages emerging independently from the primordial soup? Or did they have a more hierarchical relationship, with one domain giving rise to the others? We now know that Luca gave rise to bacteria and archaea, with these two domains diverging from each other about 4 billion years ago or slightly later. Eukarya, the domain that includes all complex cells with nuclei, emerged much later, probably around 2 billion years ago, through a dramatic event where an archaeon engulfed a bacterium, and the bacterium became the mitochondrion, the powerhouse of the eukaryotic cell. This means Luca predates the split between bacteria and archaea, but it was already a sophisticated organism with complex biochemistry. The path from chemistry to Luca must have involved many steps, many intermediate forms that are now lost to history. But Luca itself was not a primitive protocell. It was a fully functional living organism with a genome, proteins, metabolism, and the ability to reproduce and evolve. Understanding Luca helps us understand what features of life are fundamental and what features evolved later. The genetic code, for instance, was present in Luca, which is why virtually all organisms use the same code. The ribosome, in its essential form, was present in Luca. The membrane, though its composition may have varied, was present. These are the core features that define cellular life, and they were all in place by 4.2 billion years ago. Other features evolved later. Photosynthesis, the ability to use sunlight as an energy source, evolved after Luca in certain bacterial lineages. The same is true for oxygen production. The ability to respire oxygen, using it to extract energy from food, evolved after oxygen became abundant in the atmosphere. Multicellularity, the organization of cells into complex structures like plants and animals, evolved much later still, probably around 1.5 billion years ago. Each new capability, each innovation in the history of life, built on what came before. And it all traces back to Luca, the 4.2 billion-year-old microorganism that lived near a hydrothermal vent in Earth's ancient ocean, using hydrogen and carbon dioxide to power its metabolism, defending itself against viruses, and reproducing to create the lineage that would eventually give rise to all the biodiversity we see today. Luca is our ancestor in the most literal sense. Every living thing on Earth, including you, descends from that ancient organism. The molecules in your cells, the genetic code in your DNA, the basic biochemistry that keeps you alive, all of it traces back to innovations that existed in Luca or even earlier. We are, in a very real sense, 4 billion-year-old beings, carrying within us the legacy of Earth's earliest life. The discovery that Luca lived so early raises fascinating questions about life's origin. If Luca existed 4.2 billion years ago, and if life requires time to evolve from simple chemistry to the relatively complex organism Luca was, then life itself must have originated even earlier, perhaps as early as 4.3 or 4.4 billion years ago. This timeline creates challenges for our theories. 4.3 billion years ago was right in the middle of the late heavy bombardment, when Earth was being pummeled by asteroids and comets. Large impacts would have generated enough heat to boil the oceans and sterilize the surface. How could life have originated and survived under such conditions? One possibility is that life originated multiple times, being repeatedly wiped out by major impacts, and then re-emerging once conditions stabilized. Another possibility is that life found refuges where it could survive even the largest impacts. The deep ocean, particularly near hydrothermal vents, might have provided such a refuge. Water is a good insulator, and organisms living in the rock beneath the seafloor might have been protected from surface catastrophes. A third, more radical possibility is that the late heavy bombardment wasn't as severe as traditionally thought. Recent analyses of lunar craters have questioned whether there really was a sudden spike in impacts 4 billion years ago, or whether the bombardment declined more gradually. If impacts were less frequent or less devastating than previously believed, then life might have had a less interrupted path from its origin to Luca. The existence of Luca also constrains theories about the origin of life. Any successful origin of life theory needs to explain not just how life began, but how it could have evolved from simple chemistry to an organism as complex as Luca within a few 100 million years. This is a tall order, and it's one reason why the origin of life remains such a challenging problem. Yet the rapidity of life's emergence might actually tell us something hopeful. If life arose quickly on Earth, perhaps as soon as conditions permitted, this suggests that the transition from chemistry to biology, while mysterious, might not require an improbable sequence of unlikely events. It might be, in some sense, easy. Given the right conditions, liquid water, energy, and organic molecules, life might emerge readily, constrained more by chemistry and physics than by chance. This has implications for the search for life elsewhere in the universe. If life is easy, if it arises quickly wherever conditions permit, then perhaps life is common in the cosmos. The universe might be teeming with microbial life on worlds that have liquid water and the right chemistry. This doesn't necessarily mean intelligent life is common. The path from microbes to intelligence might be much harder and much longer. But it does suggest we may not be alone in the universe, that microbial life at least might be ubiquitous. Luca serves as a milestone in life's history, a known point from which we can work backward toward the origin and forward toward the incredible diversity of modern life. Understanding Luca, its genome, its metabolism, its ecology, brings us closer to answering the fundamental questions about where we came from and what it means to be alive. The story of life's origin would remain entirely theoretical, built on experiments and genetic analyses, but lacking direct historical evidence, if not for the fossil record. Fossils provide our only direct window into the deep past, preserving evidence of ancient organisms in rock. And remarkably, we have fossils of Earth's earliest life, structures that formed over 3.5 billion years ago, providing tangible proof that life existed in the distant past and offering clues about what that early life was like. The earliest fossils aren't the bones and shells that most people think of when they hear the word fossil. Those belong to complex animals that didn't evolve until much later. Earth's oldest fossils are stromatolites, layered structures created by communities of microorganisms, primarily bacteria, living in shallow water. Stromatolites are still forming today in a few places around the world, which allows us to study how they develop and to compare modern ones with their ancient counterparts. A stromatolite forms when a mat of bacteria grows on a sediment surface in shallow water. The bacteria secrete a sticky mucus that traps particles of sediment from the water. Over time, layers accumulate, with each layer representing a thin microbial mat covered by trapped sediment. The bacteria grow upward toward the light, abandoning the lower layers, which become lithified, turned to stone. The result is a layered rock structure with distinctive dome or column shapes. Stromatolites can grow quite large, forming mounds several meters across or columns reaching toward the surface like underwater pillars. The layering within them creates a distinctive pattern that's visible in cross-section. Thousands or even millions of thin laminations representing individual growth periods, day by day, year by year, slowly building the structure. The oldest generally accepted stromatolites come from the Dresser Formation in the Pilbara region of Western Australia, dating to 3.48 billion years ago. These ancient structures are found in rocks that were once the floor of a shallow sea or tidal flat. The stromatolites are made of minerals like silica and hematite, deposited by hot spring or hydrothermal fluids, suggesting they formed in a volcanic or geothermal environment. For years, there was debate about whether these structures were really biological or whether they might have formed through purely geological processes. Some rock formations can create layered patterns that resemble stromatolites without any biological involvement. How do you prove that 3.5 billion-year-old rocks preserve traces of life rather than being interesting but lifeless geological curiosities? The evidence comes from several lines of investigation. First, the structures have the right shape. They're dome-shaped or columnar, with growth oriented upward, exactly what you'd expect from bacterial mats growing towards sunlight. They show variations in thickness and waviness, characteristic of biological growth rather than simple mineral deposition. Second, at a microscopic level, the rocks contain structures that look like fossilized cells or filaments of bacteria. These microfossils are tiny, visible only under high magnification, but they have the right size and shape to be bacterial remains. Some show signs of having been organic, with chemical compositions distinct from the surrounding rock. Third, and most convincingly, recent analyses have found organic matter preserved within the stromatolites. In 2018, researchers studying the Dresser Formation found complex organic molecules trapped in tiny pores within the rock. These molecules are consistent with the remains of bacterial biofilms, the slimy mats that stromatolite-building bacteria produce. This organic matter is the smoking gun, definitive evidence that these structures are biological. The Pilbara stromatolites are currently considered the oldest definitive fossils on Earth. But there are older, more controversial candidates. Rocks from Greenland, from a formation called the Isua supercrustal belt, contain structures that some scientists interpret as stromatolites dating to 3.7 billion years ago. These would be 200 million years older than the Australian ones. However, other scientists have argued that the Greenland Structures might be geological rather than biological, formed by tectonic forces rather than by bacterial growth. The debate continues. Even more controversial are claims of microfossils from the Nouveau-Agatuque Belt in Quebec, Canada. These rocks, which formed near a hydrothermal vent on the ancient seafloor, contain tiny filaments and tubes that some researchers interpret as the remains of iron-oxidizing bacteria. If these structures are biological, they could be as old as 4.28 billion years, which would make them not only the oldest fossils on Earth, but also roughly contemporaneous with Luca. The problem with such ancient fossils is that the rocks have been subjected to intense heat and pressure over billions of years. They've been metamorphosed, their minerals rearranged, their structures deformed. Distinguishing biological shapes from geological artifacts becomes increasingly difficult the further back you go. Scientists demand extraordinary evidence for extraordinary claims, and proving that 4-billion-year-old structures are biological requires ruling out all possible non-biological explanations. The importance of stromatolites extends beyond their age. They tell us what early life was like. The organisms that built these structures were photosynthetic bacteria, using sunlight to produce energy. This means photosynthesis, one of the most important biological innovations, arose very early in Earth's history. The bacteria were also capable of forming communities, working together to build structures much larger than any individual cell. This communal lifestyle, with different bacteria potentially playing different roles, suggests a surprising level of biological sophistication even 3.5 billion years ago. Stromatolites also tell us about Earth's environment. The bacteria that build stromatolites need sunlight, which means they lived in shallow water. They need liquid water, confirming that oceans existed at least 3.5 billion years ago. The minerals within the stromatolites reflect the chemistry of those ancient oceans, which were different from modern seawater, more acidic and with different concentrations of dissolved metals. One of the most fascinating aspects of stromatolites is that they were incredibly abundant during Earth's early history. From about 3 billion to 1 billion years ago, shallow oceans around the world were carpeted with stromatolites. They were the dominant form of life, shaping entire ecosystems. Then, quite suddenly in geological terms, they declined. By the time of the Cambrian explosion, 540 million years ago, stromatolites were rare. What happened? The most likely explanation is grazing. When the first animals evolved, including simple invertebrates that could eat bacteria, they began consuming the microbial mats that formed stromatolites. Modern stromatolites exist primarily in places where grazers are rare, like the hypersaline waters of Shark Bay in Australia, where the high salt content excludes most animals. In more normal marine environments, grazers keep bacterial mats in check, preventing stromatolite formation. This decline of stromatolites coincides with the rise of animal life, providing a poignant reminder that evolution is a competitive process. The bacteria that had dominated Earth's ecosystems for billions of years were suddenly faced with predators they couldn't defend against. Stromatolites didn't go extinct, they still exist, but they were pushed to the margins, restricted to a few extreme environments where they could persist. Studying ancient stromatolites requires traveling to remote locations and examining rocks with specialized equipment. But the effort is worthwhile. These structures provide our only direct evidence of what life was like billions of years ago. They show that life didn't just survive on the harsh young Earth. It thrived, building vast structures and fundamentally altering the planet's surface and atmosphere. The photosynthetic bacteria in stromatolites were the first organisms to produce oxygen as a byproduct of their metabolism. For perhaps 1 billion years, this oxygen reacted with iron dissolved in the oceans, causing the iron to precipitate and fall to the seafloor. These precipitates formed banded iron formations, distinctive layered rocks that are the main source of iron ore mined today. Eventually, around 2 billion years ago, all the dissolved iron had been used up, and oxygen began accumulating in the atmosphere, causing the great oxygenation event that transformed Earth's environment and paved the way for the evolution of complex life. So, stromatolites aren't just passive fossils. They're evidence of organisms that fundamentally changed the planet, that set in motion processes that would eventually lead to the oxygen-rich atmosphere we depend on today. They're proof that even simple life can have planetary effects, that biology and geology are intimately connected, that life doesn't just adapt to its environment, but actively shapes it. As we study these ancient structures, we're reading messages from the deep past, learning about a world that no longer exists, but that shaped everything that followed, including us. The fossil record, fragmentary though it is, provides our most direct connection to life's history, anchoring our theories in tangible evidence and reminding us that the story of life isn't just a story we tell. It's a story written in stone, waiting to be read by anyone willing to look closely enough. We've traveled through 4 billion years of history, from the hellish Hadean Earth to the first stromatolites, from simple molecules to the complex organism that was Luca. We've explored hydrothermal vents and lightning storms, RNA worlds, and prebiotic soups. We've seen how far science has come in understanding life's origins. And yet, despite all this progress, we still don't know exactly how life began. We have pieces of the puzzle, many pieces, but the complete picture remains elusive. The origin of life is still one of science's greatest unsolved mysteries, and it may remain mysterious for a long time to come. Let's consider what we do know with reasonable confidence. We know that the building blocks of life, amino acids, nucleotides, sugars, and lipids, can form through ordinary chemistry under conditions that probably existed on the early Earth. The Miller-Urey experiment and countless follow-up studies have demonstrated this convincingly. We know that these building blocks are also present in meteorites and can form in interstellar space, so they were likely abundant on the early Earth through both local synthesis and extraterrestrial delivery. We know that membranes can self-assemble from simple lipids, creating compartments that could serve as protocells. We know that RNA can both store information and catalyze chemical reactions, providing a possible solution to the chicken and egg problem of heredity and metabolism. We know that hydrothermal vents provide energy gradients and chemical disequilibrium that could power prebiotic chemistry. And we know that life existed by at least 3.5 billion years ago, possibly earlier, based on the fossil record and genetic analyses of Luca. These are not small achievements. They represent decades of experimental work, theoretical development, and careful analysis of ancient rocks and modern organisms. They tell us that life's origin was not a miracle that violated natural laws, but a process that followed chemistry and physics, that can be studied scientifically, that can be understood, at least in principle. But many crucial questions remain unanswered. Perhaps the most fundamental is the question of environment. Where exactly did life begin? Was it in a warm little pond on land, in the open ocean, at a deep-sea vent, in the porous rocks of Earth's crust, in some other location entirely? Each proposed environment has advantages and disadvantages, and scientists continue to debate which was most likely. Related to this is the question of chemistry. What was the first self-replicating molecule? Was it RNA, as the RNA world hypothesis suggests, or was it some simpler pre-RNA molecule? How did this molecule form, and how did it transition from being a chemical curiosity to being the foundation of heredity? We can make RNA in the laboratory under controlled conditions, but could nature have done it on the early Earth without a chemist's guidance? Then there's the question of the pathway from simple replicating molecules to cellular life. What were the intermediate steps? How did metabolism begin? How did the genetic code originate? How did DNA and proteins emerge and take over roles that RNA had been filling? Each of these transitions represents a major innovation, and while we can propose plausible mechanisms for each one, we don't have a detailed step-by-step account that we can point to with confidence and say, this is how it happened. There's also the question of inevitability. Was the origin of life on Earth a cosmic fluke, an incredibly unlikely sequence of events that happened to occur on our particular planet? Or is life an inevitable consequence of chemistry, something that will arise wherever conditions permit? This question has profound implications for the search for life elsewhere in the universe. If life is common, then we might expect to find it on Mars, on the icy moons of Jupiter and Saturn, on exoplanets orbiting distant stars. If life is rare, then we might be alone in the cosmos, or at least extremely isolated. The answer to this question hinges partly on understanding the probability of the origin of life. How many potential sites for abiogenesis existed on the early Earth? How likely was it that life would emerge at any given site? These are questions that recent Bayesian analyses have begun to address, but definitive answers remain elusive. The fact that life seems to have arisen quickly on Earth suggests it might be relatively easy, but we have a sample size of 1, which makes it hard to draw general conclusions. Another open question is the role of chance versus determinism in life's origin. Was the specific biochemistry we see today, the 20 amino acids, the 4 nucleotides, the genetic code, the result of historical accidents that could have turned out differently? Or were these choices constrained by chemistry and physics, such that any life anywhere would use similar molecules and similar mechanisms? If we could rewind the tape and let evolution run again, would we get the same result, or something entirely different? We also don't know how many times life might have originated on Earth. Did it happen once, with all modern life descending from that single origin? Or did life arise multiple times, with most lineages going extinct, leaving only one survivor whose descendants include everything alive today? There might have been alternative biochemistries, different genetic codes, organisms made of different molecules that arose and then vanished without trace. Or Earth might have given rise to life just once, making the origin of life an even more singular event than we usually imagine. Looking forward, how will we solve these mysteries? Several approaches seem promising. First, continued laboratory experiments testing different environments and chemical mechanisms will help narrow down which scenarios are most plausible. As our ability to synthesize and analyze complex molecules improves, we may be able to create artificial life or self-replicating chemical systems in the laboratory, demonstrating that the transition from chemistry to biology is possible under controlled conditions. Second, studying extreme environments on Earth, places like hydrothermal vents, acidic hot springs, subsurface aquifers, will help us understand the limits of life and the conditions under which biochemistry can operate. Organisms living in these extreme environments, called extremophiles, might retain features of ancient life, providing clues about what the first organisms were like. Third, the search for life elsewhere, particularly in our solar system, could provide crucial evidence. If we find life on Mars or on Jupiter's moon Europa, we can study its biochemistry. If it uses the same genetic code as Earth life, that would suggest a shared origin, either through panspermia or through convergent evolution. If it uses different biochemistry, that would tell us that life can take different forms, that our version isn't the only possible one. Fourth, advances in genetics and bioinformatics allow us to compare genomes across all of life, tracing genes back through evolution to identify ancient features that might have been present in the first organisms. As we sequence more genomes and develop better computational tools, our portrait of Lusa and of pre-Lusa life becomes clearer. Finally, theoretical work, including computer simulations and mathematical modeling, can help us understand which chemical networks are capable of self-replication and evolution, which environmental conditions are most conducive to life's origin, and what the minimal requirements for life might be. These models can guide experiments and suggest new avenues for research. Despite all this effort, it's possible we'll never know exactly how life began. The events occurred 4 billion years ago, and the geological record from that time is almost entirely erased. We're trying to solve a cold case with fragmentary evidence and no direct witnesses. But even if we never achieve complete certainty, we can continue to narrow the possibilities, to rule out scenarios that don't work, to refine our understanding of what must have happened. And perhaps that's enough. Perhaps the origin of life will always retain some mystery, some uncertainty, reminding us of how much we still have to learn about the universe and our place in it. The fact that we can ask these questions, that we can design experiments and analyze data and build theories, is itself a testament to how far life has come from those first simple molecules in Earth's ancient oceans. We are, after all, the universe trying to understand itself, star stuff contemplating stars, life investigating its own origins. And in that investigation, in that quest for understanding, we're continuing the journey that began 4 billion years ago, when chemistry became biology, when molecules became life, when the first spark ignited on the young Earth. The mystery remains, but we're getting closer to solving it. One experiment, one fossil, one genome at a time. And even if we never find the complete answer, the search itself enriches our understanding of life, of Earth, and of the cosmos we inhabit. As we look to the future, new technologies promise to shed more light on life's origins. Advances in microscopy allow us to examine ancient rocks at unprecedented resolution, potentially revealing microfossils too small or too subtle to detect with earlier methods. New mass spectrometry techniques can identify organic molecules present in tiny quantities, helping us distinguish biological from non-biological materials in ancient rocks. Space missions are exploring other worlds in our solar system, looking for environments where life might exist or might have existed in the past. Mars rovers analyze rocks for signs of ancient microbial life. Future missions will return samples from Mars for more detailed analysis on Earth. Spacecraft are being designed to explore Europa and Enceladus, icy moons with subsurface oceans that might harbor life. The search for life beyond our solar system is also accelerating. Telescopes can now detect the atmospheric compositions of planets orbiting distant stars, looking for biosignatures, gases like oxygen and methane that might indicate biological activity. In coming decades, more powerful telescopes will allow us to search thousands of exoplanets for signs of life. Each of these efforts, whether in the laboratory, in Earth's rocks, or in space, contributes to our understanding of how life begins, and perhaps someday, these pieces will come together into a complete picture, a satisfying answer to that most profound of questions: how did life begin? And so we've reached the end of our journey through deep time, from the formation of Earth to the emergence of the first life, from simple molecules to complex cells, from chemistry to biology. We've seen how far science has come in understanding this profound mystery and how much remains unknown. The origin of life is one of those questions that connects us to something larger than ourselves. It reminds us that we're part of an unbroken chain stretching back billions of years, that every atom in our bodies was forged in stars and circulated through ancient oceans, that the DNA in our cells preserves information that's been copied and passed down through countless generations, all the way back to Luca and beyond. It's humbling to realize how much had to happen for us to exist. The planet had to form. The conditions had to be just right. Not too hot, not too cold, with liquid water and the right chemical ingredients. Molecules had to organize themselves into replicating systems. Those systems had to evolve, becoming more complex and more capable. 4 billion years of evolution, of trial and error, of extinction and innovation, led from those first protocells to the diversity of life we see today, including intelligent beings capable of contemplating their own origins. And yet, for all its improbability, here we are. Life found a way. Chemistry became biology. The universe became aware of itself. As you drift off to sleep tonight, perhaps you'll dream of that ancient Earth, of lightning illuminating toxic skies, of hot springs bubbling in volcanic landscapes, of deep-sea vents ejecting mineral-rich water into cold oceans. Perhaps you'll imagine the first replicating molecules, struggling to copy themselves in a hostile world. Perhaps you'll envision Luca, that ancient microorganism living near a vent 4 billion years ago, the ancestor of everything alive today. And perhaps you'll feel a sense of connection, a recognition that you are part of this story, that your existence is the continuation of a process that began when Earth was young, that every living thing shares a common heritage reaching back to life's earliest days. If you enjoyed this exploration, if these ideas gave you something peaceful to think about, something to ponder as you fall asleep, please consider liking this video and subscribing to the Quiet Science channel. There are so many more stories to explore, so many more mysteries to contemplate, so many more journeys through deep time and vast space. Thank you for spending this time with me. Sleep well, dream deeply, and remember that you are, quite literally, made of star stuff, carrying within you the legacy of 4 billion years of life on Earth. Good night.